Hold on, I had my speakers off. There we go. That sounds like that's working. All right, excellent. So let's go ahead and get started. Today is Tuesday, July 6th. I, because of the long weekend, was able to furiously work at the grading and got the exams done. Lab and lecture exam 2 have been graded. Your scores have already been posted, and then today after class, I will release the exams for review. I strongly encourage you to take a moment to review them. Take 2 moments even to review them. They'll be available for the next 24 hours, and then they'll be locked again, at which point you can still view them if you like. You just have to come to my office hours to do that. The averages, again, were not great. We have a fair number of people who did very well, a fair number of people who did poorly. The good news is your grades in this class are not solely determined on your exam scores. It's all your points together, and we've crossed the halfway point in points, and the average is 75 for the class overall, which is not bad, but I definitely would like to see that come up. However, the one thing that I do want to address for the exams is that despite the numerous times that I have said them, there are 3 main issues that I see again and again and again. Again, as I've mentioned many times, you want to make sure that you read the questions carefully. There are lots of examples of people who did a great job of describing to me the structural classifications of joints when I asked for the functional, or did a great job of describing to me how bones grow within cartilage when I asked for intramembranous ossification. Make sure you read the questions carefully so you can answer them correctly. Secondly, despite my numerous warnings, make sure that you answer all parts of a question. If I ask for a bone and a bone feature, don't just tell me the occipital bone, or don't just tell me the conoid tubercle. Make sure you're giving me all the parts. If an essay question has 3 parts, make sure you're answering all 3 parts. If a comparative question asks for 4 examples, make sure you're giving me 4 examples. Again, these are the types of errors that are very, very easy to fix. And as long you should have plenty of time on the exams to be able to do this properly, especially the lecture exams. So there's really no excuse for making these errors. These are things that you should be able to very quickly and very easily resolve. The third issue, and the thing that I probably wrote the most on this exams, is to be descriptive. Right? Give me details. That's something that we can work on. And in fact, that's one of the things that we will emphasize today on how you can be more descriptive in explaining a process to make sure that you give me the details that are going to maximize your points on that exam. So the first 2 things are easy things that you should easily be able to quickly fix. The last one, this being descriptive, is something that we'll really emphasize and work at today when we do go through today's lecture and today's big processes that we're going to be talking about. And that does bring me to today's schedule. Again, what we have for a schedule today is going to be the schedule basically for the next 5 lectures. We in lecture are going to do our muscle physiology. We have broken everything down to the most basic components, all the anatomy, all the way down to those proteins. And now we're going to put those proteins together and talk about how they work. And then in lab, we'll be focusing on the origins, insertions, and actions for your muscle list, getting everything that you need to help you to prepare for your lab exam. Again, fair number of assignments coming up. Thursday, I happened to notice that there wasn't an assignment, and being the smart, sophisticated student that you are, anytime we don't have an assignment or something like that, that's always a good day for a daily quiz. So again, you never know about those types of things. Oops, I don't need that up. Let's go ahead and close my mail. Don't need that making noise all day. Friday, your Unit 10 review is going to be due. Next week, your first part of the week, Monday and Tuesday, you have 3 assignments due. Your PhysioX exercise 2 that has 7 activities. And again, as you've seen, the PhysioX is not hard, it's just time consuming. The last one had 5. This one has 7. So make sure you do all the activities, get all of the lab reports, print the lab reports as PDFs and save them as PDFs and post those to the assignment site. And then on Tuesday, your Unit 11 review and also your muscle activity handout is due. It's going to be graded for correctness. You then get a day off, your 1-day weekend, and then we have our lab and lecture exam. This is also a reminder that this weekend is your big weekend to study, where you have 2 days off to be able to do that, and then the 1 additional day before the actual exam. So we are rapidly approaching the next section because everything is rapid in summer school. All righty, questions on any of that?
Just a question about the muscle activity handout. Is there a way that we could do it like the bone where it's due on the Wednesday so we get the lecture on Tuesday?
No.
Okay.
Because I think for this one, again, because it's graded for correctness, I'm like I was able to do for the 30 points, I was able to grade them and post them beforehand, and post the key and all that kind of stuff. This one is more of a fun activity. It's more subjective. Again, because there isn't just one right activity that you'll be able to do. Like I talked about for the bicep brachia, I use the example of bowling. There's plenty of other examples, right? If you're recently engaged, right, and you want to show it off, you are going to flex your shoulder and flex your elbow and supinate your hand so you can show off the ring. I mean, there's no wrong answer to these. Well, there are wrong answers, but there's no just one correct answer to this. I want you to be creative. I want you to be creative in the way you come up with these, because the point of this is really to make a study guide that's going to help you to be successful with this information. So sooner, I think, is better than later for something like this one. Okay.
Sounds good.
All righty, excellent. Plus, also, I don't think that the stuff we discuss in class is necessarily as important. You know now what a flex is and an extension is, and an abduction and an adduction and those types of things. So I don't think we have to necessarily go over them all together the same way that we do with the bone features. So I think a flexion of the knee is a flexion of the knee, and I don't think that we need to necessarily do that together to figure out that activity. Right, we will be going over the attachment points, and that's something we definitely need to go together. But the actions should be something that should be much more intuitive. But I appreciate you asking. All right, any other questions? All right, excellent. So let's go ahead and get started with the lecture, picking up where we left off. Oh, and actually, now that I've got the lecture up, I've changed my mind. Let's actually start by doing it this way. We left off the last class, and we talked about process of a muscle contraction. And we also talked about how this process of a muscle contraction, we are going to artificially, but not randomly, divide this entire continuous process into 3 steps. So I spell steps correctly. Perfect. Excellent. So what was the first step in that process?
Communication at the neuromuscular junction.
Excellent. And anytime we identify one of these steps, one of the things that can help us is also to think of it in terms of what the goal is of these steps. So in communication at the neuromuscular junction, because this isn't just a random dividing of this singular process into 3 steps, what was the goal of our communication at the neuromuscular junction?
To convert a neural action potential into a muscle action potential.
Excellent. Excellent. All right, we want to take the language of our nervous system, that neural action potential that is used to spread down our neurons, spread the meter from the small of our back down to the tip of our toes, and so on and so forth, and use it to stimulate our muscle cell. Remember, one of the characteristics of our muscle cell is that they're irritable, they're electrically excitable. When they receive a stimulus, they respond, and they respond by producing an electrical signal. And that is the muscle action potential. Excellent. Now, one of the other things that I will do this at the end. What was step 2?
Excitation and contraction coupling.
Excellent. Excitation, contraction, coupling. And of course, we always want to think in terms of what is the goal. What is our goal here?
To trigger with the calcium, the myosin.
Excellent. Okay, so you got the right idea. We want to use calcium to do something. You're right. We want the myosin and the actin to interact. But remember, myosin always wants to interact with the actin. So why isn't it interacting with the actin all the time?
It's being inhibited.
By what? Oh, there you go. Excellent. Samuel's got it. The regulatory proteins, absolutely. Sorry, Samuel, I didn't see that at first. Excellent. We want to use calcium to move the regulatory proteins. Excellent. All right, we have those regulatory proteins, the troponin and tropomyosin, and we're going to move calcium, use the calcium to move those out of the way. And that will allow for step 3. And step 3 was what?
Contractile cycle.
Excellent. Contractile cycle. We can also call it the sliding filament theory. As we will see, the actin and the myosin are going to slide over the surfaces of each other. And so that's where it gets that name, the sliding filament theory. But I actually like the term contractile cycle a little better because it reminds us, like all cycles, that it is a continuous process. So it isn't something we're going to just go through once. It is something that is going to continue to go around and around and around as long as the conditions are appropriate for that. Excellent. And then, as we mentioned, if there was a 4th process, what would that 4th process be?
Muscle relaxation.
Yeah. Or we could just simply think of, when we talk about all the processes, we can talk about initiating it and terminating the process. So we could think of relaxation as its own separate step. Or for each of these steps, we can talk about how we're going to initiate it and then how we're going to terminate it. Now, the other point that I want to remind you is, again, muscle contraction is a continuous process, right? These steps are artificial. We're breaking it artificially. And so because of that, one of the important things to remember is the end of step 1 is also going to be what?
Let's say
it this way. The very last step of part 1 is also what?
The very beginning of step 2.
Yeah. And then, so by the same token, the very last step of part 2 is the very first step of part 3. There you go. because this is a continuous process, it's going to be easy to remember that exactly where 1 ends is exactly where 2 is going to pick up. Exactly where 2 ends is exactly where 3 is going to pick up, because this is a continuous process. All right, that is our game plan. That is our goal. Now, there is a little bit more anatomy we need to talk about. So any questions on this before I erase this and we move on to the next thing? All right, excellent. Our focus now is going to be on step 1. Or part 1, let's go with part. Communication at the neuromuscular junction. And again, anytime we're doing this, we should also think in terms of their goals. What was the goal again? Come on, we literally just did this.
To convert the neural action potential to the muscle action potential.
Oops. So again, we have to go from the language of the nervous system to the language of the muscle system. Convert that signal. And to do that, we need to know a little something about the nervous system. As we know, skeletal muscle is controlled by the somatic motor nervous system. And so we need to talk some very basic anatomy about a somatic motor neuron. Like most cells, a neuron has a big, large, centrally located body. And of course, in that cell body, it's got most of the stuff we expect to see inside of a cell, like a nucleus, like the rough endoplasmic reticulum with the dots all over it, and smooth endoplasmic reticulum, and a Golgi apparatus, and all the other stuff we expect to see inside of a cell. But one of the big specializations of neurons is neurons have processes. Processes is a term we are familiar with because we just finished talking about bones. And bones have processes, and a process is something that sticks up off of it. And in the case of neurons, neurons basically have 2 types of processes. The first of these are small, typically elaborately branched processes. And these small, elaborately branched processes are called dendrites. And let's cheat. We'll move the cell body in here. Dendrites are small, branched, and they receive information. But neurons also have one big, large, unbranched process. And this one big, large, unbranched process is called an axon. As I mentioned, you have neurons whose cell bodies are located in the small of your back. And they have to reach all the way down to the tip of your toe, which is like a meter long. So these axons can be very, very long. They're unbranching. And their job is to send information. And they send that information in the form of an action potential. And an action potential, well, and let's be specific, neural action potential. And like all action potentials, this is a big positive signal. In fact, the average action potential is about a 100 millivolt change, positive signal. Big, huge, massive signal. Now, eventually, that neuron is going to reach whatever its target cell is. And when it reaches that target cell, something special happens to the axon. The axon actually starts to branch. And at the end of those branches is a specialized structure where it will communicate with the target cell. Now, this is not drawn to scale at all, but I'm just doing this to emphasize that. So here we have our target cell. Here we have these specializations to our axon. Now, I want to emphasize that depending on the branch of neuroscience, depending on psychology, there are literally dozens of names that are used for these. And you, of course, are welcome to use any of these. The ones that I will use standardly for me, just to, again, try to avoid confusion, is where the axon branches are what are known as the axon terminals. And the specialized structures at the ends are what are known as synaptic end bulbs. And it is here at the synaptic end bulbs where our neuron communicates with the target cell. And this special region of communication is called a synapse. This is the generic term or general term. Now, how many different types of cells in the body do neurons communicate with? How about lots?
Lots.
They communicate with other neurons, they communicate with glands, they communicate with skeletal muscle, cardiac muscle, smooth muscle, right? All sorts of different things can be targets for our neurons. So synapse is the generic term, the general term for this area of communication. However, the area of communication, the synapse, between a somatic motor neuron and a skeletal muscle cell has a special name. And what do you think we call that special synapse located between or formed by our somatic motor neuron and our skeletal muscle cell? There you go, exactly. The neuromuscular junction. So the neuromuscular junction is a synapse. All right. It is the area of communication between our somatic motor neuron and our target cell. And of course, in this case, our target cell is a skeletal muscle fiber or cell. All right. Questions on this?
Nope.
Excellent. All right. So this is the simple overview. Obviously, we'll get a lot more when we talk about the nervous system. But notice what we care about is this right here. The area of communication between the neuron and our skeletal muscle cell. So what we're going to do is we're going to blow that up and we're going to take a look at it. So let's erase all of this. And we are going to focus on oops, don't need this to be capitalized. We're going to focus on the anatomy of the synapse known as the neuromuscular junction. And it has 3 parts. Part 1 is the neural part. And that neural part, as we've talked about, is the synaptic end bulb. Again, these are called synaptic boutons, terminal buttons, synaptic buttons and boutons. Again, all sorts of different names that they have for them, but we're using the term synaptic end bulb. And we can draw it. This would be the axon terminal coming down, and this would be the enlargement. That is our synaptic end bulb. So again, this is my axon terminal. And this is my synaptic end bulb. Now, my synaptic end bulb is a specialized structure for communication. Now, we know our neuron produces a big electrical signal. We call it the action potential. But it turns out it doesn't spread that action potential directly to the muscle cell. They're not hardwired together. Instead, it turns out the way it communicates with the target cell is by releasing a chemical. So one of the specializations here of our synaptic end bulb is it contains a large number of vesicles. These vesicles, oops, that's not right, contain a chemical signal for communication. Now, there is, of course, a generic term for this chemical used for communication. Does anyone know what the generic term for chemical that a neuron uses and releases for communication might be?
Acetylcholine.
Well, you're correct. Acetylcholine is the specific chemical that is released, but Arabella's got it. The generic term is a neurotransmitter. Over 100 different neurotransmitters have been identified in the human body. But here in the neuromuscular junction, where our somatic motor nervous system communicates with a skeletal muscle cell, there is one and only one specific neurotransmitter that is used. And the specific neurotransmitter, as you mentioned, is acetylcholine, which after you've written it out once on an exam, like in an essay question, you can abbreviate ACH, although capitalize the A. There we go. Excellent. So there you guys go. Together we got it. Here inside, our vesicles are that neurotransmitter, that chemical that it is going to use for communication, called acetylcholine. Now, we will talk about the process, but when the neuron releases acetylcholine, that is going to cause our muscle cell to contract. However, as we've talked about, do we want our muscle cell contracting all of the time?
No.
No. So we need to have a way of controlling the release of this neurotransmitter. And releasing neurotransmitters is kind of a wonky thing. So what would be a good way to make this cell do a wonky thing like releasing its neurotransmitter? Use calcium, exactly. So what this cell is going to have on it, and I'll put a couple up here, and I'll put a couple up down here, and then we'll talk about what they are. What this cell is going to have is it is going to have a special channel that is going to let calcium into the cell to release this neurotransmitter. However, we don't want it letting calcium into the cell all the time. We don't want to be releasing acetylcholine all the time. So remember, we said we could make channels that could open and close, and we called them gated channels. Gated channels are doors that can be locked and unlocked. And of course, if something can be locked and unlocked, it needs a key. And in this case, the key is a change in voltage. As we know, what is the resting membrane potential of a typical cell?
-70.
Negative 70 millivolts. However, if we can change that resting membrane potential, we can unlock that voltage-gated calcium channel, and we can allow calcium into the cell. So we have our neurotransmitter to be released. We have our voltage-gated calcium channels to control that release. And it turns out this process requires a massive amount of energy. So not surprisingly, the other thing we find inside here is a large number of mitochondria. So we'll just throw some mitochondria in there as well. This is a very active process. We have lots of mitochondria. There we go. That is basically our synaptic end bulb and the key things we need to know about it. Now, we mentioned our neuromuscular junction has three parts. One part is the neural part, the synaptic end bulb. The other part is the muscle part. Oops, I didn't mean to do that. As we know, a skeletal muscle fiber is a long cylinder-shaped cell that can be up to a foot long, and it has that special sarcolemma on the outer surface. But on one specific part of that sarcolemma is a structure called the motor end plate. So we'll cheat real quickly up here. We know a muscle cell is this striped cell, can be up to a meter long, cylinder in shape, nuclei all over the place. But what happens, and we'll just do it this way. In one little location, so just like right there, is a region called the motor end plate. And again, just like the synaptic M bulb, this is a region that is going to be specialized for communication. Now, one of the ways it is going to be specialized is by having a large surface area. After all, our neuron is releasing a chemical signal. So much like that English muffin you had for breakfast this morning, the more nooks and crannies it has in it, the more butter it can capture. Well, it's the same thing here. By having all these nooks and crannies in our motor end plate, these invaginations, as we know, these invaginations increase our surface area for both more contact and to retain the acetylcholine within those spaces for longer. So it can have a bigger effect. Well, and how does that chemical have an effect? Because on the surface of this, we have some more channels. And I'm going to cheat, even though the last one was a channel 2, I'm just going to draw these channels as blocks. These are channels. And these channels allow cations to flow through them. What is a cation again?
Positively charged ions.
Positively charged ion. And what are the two most common ones associated with the body and interstitial fluid in the cell itself?
Calcium.
Calcium is an important one, but it's not the most common.
Sodium and potassium.
Potassium.
There you go. Sodium and potassium. There are some other ones, magnesium, a little bit of calcium that can sneak in, things along those lines. But primarily when we talk about things that flow through this cation channel, it is primarily sodium and potassium that are going to flow. All right. Now, again, this one is one that the chemical is going to bind to. So that, see, now that I've done that, I'm going to rewrite this. Our acetylcholine can bind to the surface of these, and that is the key that unlocks the door. So let's go ahead and get rid of that. So these are also a gated channel, but in this case, they're chemically gated, or what the books sometimes refer to as ligand gated. A ligand is just a molecule. So chemically gated, ligand gated literally mean the exact same thing, but you may see the term ligand gated, and I don't want you to be confused by that. So we have a chemically gated cation channel that, as we know, will primarily let potassium and sodium flow. So our specializations to the motor end plate are this big invagination and these chemically gated cation channels. Now, remember also, I'm going to grab this, put it down here so it's out of my way. Actually, I'll stick it there so it's, I think it's out of my way enough there. Notice I said that a neuromuscular junction has 3 parts. The third part, as it turns out, is actually the space in between. So the third part is actually a space. The two cells don't actually touch. And of course, this space has a name, and that name is the synaptic cleft. Our two cells are very close together. If you think about it, and we've talked about this before in this class, it would actually make sense for these two to touch. If the two touched, if they actually had those gap junctions that connected them, then when our neuron fired an action potential, our muscle cell would always fire an action potential, and we would always get the activity that we want. It would be fast, it would be convenient, it would be really useful. Why don't we have these two cells touching? Why not have that gap junction? What's the purpose of not allowing them to touch?
Because the muscle's going to contract.
But isn't that what we want? Don't we want the muscle to contract?
But it's going to move.
Well, but don't we want it to move? Isn't that the point of stimulating the muscle cell, so that it contracts, so that it moves? I'm sorry, you broke up at the beginning of that. What?
It's going to contract indefinitely.
Oh, I see what you're saying. Okay, yes. If the neural cell continued to produce action potentials, then the muscle could continuously contract. But we don't have to continuously produce action potentials. We can hardwire these things together. If these two things were connected and hardwired, then just like the light switch in your house, every time you flipped on the light switch, the light goes on. Every time you flip it off, it goes off. So we could turn it on and off, and we would have, right, by hardwiring it together, we'd get that fast, rapid, always response, which would be awesome, wouldn't it?
You asked the question, so I'm going to say no.
Exactly, right? While it is useful to have things hardwired together, remember, as we talked about, we need to be able to modify our behavior. Because while that light switch gets turned on, the light always goes on. We don't necessarily always want to have a response. If I stand here with this, sit here in my chair on Zoom with you, with a red pen in my hand, look you in the eye through my camera and say, I'm going to kill you, and a bear with an axe walks into your room, looks at you in the eye and says, I'm going to kill you, you're getting the exact same stimulus. Do you want to respond the same way to both of those?
No.
No, absolutely not, right? And even reflexes, right? There you are sitting in your doctor's chair, and he takes the little hammer and hits you in the knee, and what happens when he hits you in the knee?
A little reflex kick it.
Yeah, your leg kicks out as a result of that. Absolutely. Excellent. So then he takes that hammer, you're sitting there in the chair, but he puts a sleeping baby right next to your foot. And then he hits you in the knee with the hammer. Does your leg fling out kicking that baby?
No.
No, right? You're able to modify your behavior. And that's why we don't have these things hardwired together. By not having them hardwired together, we can modify our behavior. And that's the big, huge difference with these synapses, right? So these two cells are very close together, but they do not touch. They are not hardwired together. There is a space in between. Now, of course, this space isn't just void, it's filled. And of course, this space, being outside of the cells, is filled with interstitial fluid. Right? That's important because, as we know, is there a lot of sodium or very little sodium in interstitial fluid?
Lots.
Lots. A lot of potassium or very little potassium in interstitial fluid?
Little.
Little. Excellent. But there's also some special proteins in here as well. So it's filled with the interstitial fluid, but also contains some special proteins. In particular, one of those proteins is a protein called acetylcholine esterase, which again is an alphabet soup term. I guarantee you're going to have to spell at some point or another on the exam. But the good news is once you've written that out once, you can abbreviate it A, capital A, CH, capital E. And as the name ending in ACE indicates, this is an enzyme, a little Pac-Man. And this Pac-Man's job, as we know, is to eat dots. In this case, what are the dots?
Acetylcholine.
There you go. Acetylcholinesterase breaks down acetylcholine. So we have the special enzyme in here that can help in the breakdown of our acetylcholine. And there you go. This is our synapse. Notice oops, hold on, wrong button. I meant to save that, not delete it. Here we see a low magnification view. Notice, there it is. Here's our axon coming down. Here's our branches, the axon terminals, and our synaptic end bulbs at the end of this. Again, this is our neuromuscular junction, a very specific type of synapse. And remember, synapse is the general term that we use for an area of communication between a neuron and its target cell. Dozens upon dozens of different types of synapses have been found in the human body, but right now we're just focusing on one of them, the neuromuscular junction, that communication between our somatic motor neuron and our muscle cell. And as we've seen, if we take a closer look, that synapse has three components. The neural component, which is the synaptic end bulb. Notice as we talked about that synaptic end bulb, here, let's actually use the highlighter, has the vesicles containing our neurotransmitter, the chemical signal used for communication. And in this case, that is acetylcholine. I think I have that there. Okay, so yeah, we'll come back to it. It also has the specialization of the voltage-gated calcium channels that is going to allow calcium in, that's going to trigger the release of that acetylcholine. And like I said, lots of mitochondria because this is an expensive process. We have our specialization of the muscle cell, our motor end plate that has these big, huge invaginations to increase its surface area to allow for more contact with our neurotransmitter. And then, of course, our chemically gated cation channels that the acetylcholine will bind to and allow sodium and potassium to flow. And of course, the two don't touch. There is that space, that fluid-filled space in between known as the synaptic cleft, where we also have some special proteins like our acetylcholine esterase that can break down our acetylcholine. Again, the way our neuron communicates with its target cell is by releasing a chemical signal called a neurotransmitter. And at the neuromuscular junction, it is acetylcholine. In fact, acetylcholine is our most common neurotransmitter in the peripheral nervous system. So we're certainly going to talk about acetylcholine now, but there will be other times when we talk about it as well. All right. Questions on that?
Yeah, I had a question.
Yes.
What's keeping the axon in place?
Hope.
Hope.
Yeah. No, great question. There are a couple things. The first is let's go back to this. Most axons tend to run parallel to blood vessels. So blood vessels, nerves, and axons and arteries tend to, veins, arteries, and and nerves tend to travel together. And like most of the things in the body, just like we saw our muscle cells, a lot of connective tissues are going to hold them together. So these are things that are going to be anchored in place by connective tissues that surround them and support them. Yep. And just like most muscle cells don't hang out by themselves, most axons don't hang out by themselves. They're actually bundled together, and we actually have a special name for a bundle of axons. What do we call a bundle of axons? Anyone remember? I know you know, even if you don't know, you know. My kids get on mine all the time.
Nerves.
There you go. A nerve is a bundle of axons, and they're bundled together and supported and connected by connective tissues that also anchor them in place as well. Yep. Great question. Any others?
Yes. So we're looking at one axon and one muscle fiber. Now, is there multiple of these and just hitting all of these at one time, or is it just kind of like it does
Okay.
one and it communicates with the rest?
So great question. Here's what I would say is to remember that we have broken everything down to its most basic component. And we do have to build up to understand how we move our body through space. Right now, we are focusing on the communication with one muscle cell. And what I will tell you is that one muscle cell has one and only one motor end plate. So each muscle cell has one and only one neuromuscular junction. And so therefore only receives input from one somatic motor neuron. So yeah, so each muscle cell gets input from one and only one somatic motor neuron. Now, it's jumping ahead a little bit, but does each somatic motor neuron only communicate with one muscle cell? No, right? Every soldier has one general who gives him directions, but does each general only control one soldier?
No.
No, and it's the same way here. One somatic motor neuron can control thousands of muscle cells, but each muscle cell only has one neuromuscular junction and only gets input from one somatic motor neuron. All right. Good questions. Any others? All right, excellent. With that, our goal then is going to be to talk about this first step. Go through all of the processes through the first step. I will be completely honest with you. The next hour and change of lecture is my absolute favorite lecture in the entire 430, 431 series. This is my favorite part of the class to give. And so I'm stoked, and you should be as well. Lot of material, ton of material we are going to go over. So even though it's a little bit early, this is when we're going to take our first break. Let's take a full 15-minute break, and that brings us back at 9:10. And do whatever you need to do to get ready. Go get a cup of coffee, drink a Coke, do a rail of Coke, take a power nap, whatever it is you need to do to be ready, do that to be ready, because when we come back, I am taking you on a wild ride. All right, any questions? All right, see you in 15 minutes. All righty. As we've talked about, our goal now is to continue our discussion. And again, we are going to talk about part 1, communication at the neuromuscular junction. And again, our goal is to convert a neural action potential into a muscle action potential. I'm going to abbreviate that just so it fits here at the top so we can keep it. Oh, that was what I was afraid of. Out of the way. And obviously the first thing we need to do for this is to discuss the anatomy. We just did this, but again, we need to put all the pieces together. Just like we've done in the past, we'll go through this once together, and then we'll go through it with all the pretty pictures and all the pretty words. So again, as we know, this starts with, way too big. Oops. I'm going to change my orientation a little bit on this so that it'll be easier for me to work and write around. So again, this is my axon terminal. And this in here is my synaptic end bulb. And someone again remind me of the specializations of the synaptic end bulb that allow it to do its job.
You talk about the channels?
Sure, that'd be one example, a channel, absolutely. So let's put a channel on it.
And the vesicles.
Yeah, well, it's okay, absolutely. So the vesicles are the other ones. But let's be more specific. Again, one of the things that gets us in trouble on the exams is not having enough detail. So absolutely, if I was describing the anatomy of a synaptic end bulb and I said it had a channel, that would be accurate, but I want to be more specific. What type of channel does it have again?
Voltage gated calcium channel.
Excellent. So they're voltage gated calcium channels, and absolutely it has vesicles. And those vesicles contain what?
Acetylcholine.
Excellent. Which is a specific type of what again?
Neurotransmitter.
Neurotransmitter. Excellent. So let's go ahead and excellent. We know there's mitochondria in here, but I'm not going to bother drawing that because we don't need to worry about that for right now. We also know that we have our muscle cell, so we'll put the muscle cell down here. Don't like that. Put the muscle cell down here. But as we also know, that muscle cell has a specialization to it, a couple specializations. What is one of the specializations to our muscle cell?
The motor end plate.
Excellent. What's special about the motor end plate?
Invaginations to increase the surface area.
Excellent. So we need to give it all its nooks and crannies. So there's our invaginations of that. And what else do we have? So what else? So we have an increased surface area, do those invaginations. And what's the other specialization?
The chemically gated cation channels.
Excellent. So we'll draw a couple of those. And these are indeed our chemically gated. Chemically gated cation channels. Excellent. So this, of course, is our motor end plate. Excellent. Oops. And there is one more specialization. What's the third specialization?
Synaptic cleft.
Exactly. We know, and I'll cheat and do it this way. Our third specialization is the synaptic cleft, which of course is a space in between, the two don't touch, contains interstitial fluid. But what's the other thing that can be found here inside of our synaptic cleft that we talked about?
High levels of sodium, low levels of potassium.
True, but
and-
that's in all interstitial fluid. So you're absolutely
Yeah.
correct, because we know the composition of it because it's interstitial fluid. But we also said there was something special in here, a special protein that's going to help us in this process.
Acetylcholine esterase.
Excellent. So we can't forget about Pac-Man here. There we go. We also have our special enzyme.
Oh yeah, I was actually going to ask with the with that enzyme, is it just going to grab the acetylcholine after it affects the channels, or is it just going to grab them randomly?
So it is an enzyme. Remember, the action of enzymes is basically due primarily to the kinetic energy of the movement of molecules. So if it bumps into acetylcholine at any time it bumps into an acetylcholine, it is going to gobble that acetylcholine up. So it doesn't know whether the acetylcholine has bound to a channel or not, and quite frankly, it doesn't care. If it sees an acetylcholine, it's going to gobble it up. Right? Break it up.
And is it conformational change? Like, is it grab it, do something with it, and release it, or it just completely eats it?
No, it breaks it down into its basic components. And so it doesn't destroy it. It just splits it, which for our purposes means it can no longer be a functional key. Right? You break that key, it can't open the door anymore. And we'll actually see how exactly this works and why this is important. All righty. So there you go. Really, if you think about it so far, no new information that we've put on the board here. This is all stuff that we have talked about already. We've reoriented it a little bit from top to bottom instead of left to right, but there's really no information, but this is still good. This is a starting point. This is our anatomy. All right, so any questions on this? Because if this doesn't make sense, it is only going to get worse from here. All right, excellent. So, as I mentioned, one of the key things we are going to need to be able to do and explain and understand is turning this process on. Of course, we want to be appropriate with the terms of this, so we want to be talking about initiation of our communication at the neuromuscular junction, right? One of the possible essay questions that I guaranteed you is going to be worded that way. Describe the process of initiation of communication at the neuromuscular junction. All right, so this is our starting point. We are now going to start talking about our first process. Excellent. And as we know, a muscle contraction starts with what?
With the neuro action potential coming down the axon.
Even before that. If I'm going to move my finger, where does that begin?
The brain.
Depending on what hemisphere.
Well, yeah, if it's my right hand, then it's at my left, but you got the right idea. It starts with the decision. Remember, this is a voluntary act. So it starts with the decision. You're absolutely correct. We make the decision that we want to move the muscle. Then, Brian, you were correct. From that, that causes us to produce that big positive signal that is an action potential. One of the things we'll learn, and we'll talk about this more when we get to nervous system. And actually, I've written it out once, so I can cheat and abbreviate now. A neural action potential is a big positive signal. Electricity moves very much like water, right? Again, being a college professor, as you know, I make major scratch. So, of course, in my backyard, I have a huge Olympic-sized swimming pool. And because I also have many, many cars, I could easily just stand at the side of my massive pool, holding one of those cars over my head and dropping it into the edge of my huge, massive pool to produce a huge, massive wave. And when I produce that huge, massive wave by dropping a car into my pool, does that wave just stay there at the edge?
No.
No, it is going to spread down the surface of the pool. And that's what happens here as well. We produce this big positive signal, this neural action potential, and it spreads down the axon to, and actually, you know what? This is going to be way too big. I'm going to have to start making this smaller. To the synaptic end bulb. All right. Now, as that wave spreads across my pool, it splashes things and gets things wet. As this wave spreads down the axon, it makes things more positive. Remember, we said a neural action potential is about a 100 millivolt change. And so as it spreads, it makes things more positive. And we have a fancy name for it when we make a cell more positive. Does anybody know that fancy term we use when you make a cell more positive?
Depolarization.
Exactly. It depolarizes the cell as it goes. And so that big positive signal comes down here to my synaptic end bulb, and my synaptic end bulb depolarizes. Now, what was its starting point again? What was the resting membrane potential?
Negative 70 millivolts.
Excellent. So our starting point was negative 70 millivolts. And we have these voltage-gated calcium channels that are going to open when our voltage changes. Now, that big positive signal that spreads down here and depolarizes, oops, hold on. I need to erase that now. When it depolarizes, it gets more positive. Do you think going from negative 70 millivolts to negative 69 millivolts is going to be a big enough change to open that voltage-gated calcium channel?
No.
No. Negative 68? Negative 67?
No.
No. But is there going to be a special point where when it gets positive enough, it's going to cause them to open?
Yep.
Yeah, and we have a special name for that. That special name we have is called threshold. Threshold is the voltage at which a voltage-gated channel opens. And it turns out in most cases, threshold is about negative 60 millivolts. So notice we only need about a 10 millivolt change, and we have a 100 millivolt wave coming. So is that going to be enough to get it to reach threshold?
Yeah.
Absolutely. Now, what is the immediate effect of this synaptic M bulb reaching threshold?
The gaze opening up.
Exactly. Our voltage gated calcium channels open. And when our voltage gated calcium channels open, hold on. And when our voltage gated calcium channels open, what happens?
Sodium wants to get in.
Sodium wants to get in?
Yep.
Calcium comes in, absolutely. Is there an imbalance in calcium inside and outside the cell?
Yeah.
Yeah, there's much more calcium outside than inside, so calcium enters the synaptic M bulb. And so our calcium comes in. Now, as we've talked about, calcium makes cells do wonky things. Do cells want calcium coming in all the time?
No.
Do they want calcium hanging out inside of them?
No.
No. So as it turns out, there's one more anatomy thing we've got to add here. And that other anatomy thing we need to add here is a calcium pump. Like all pumps, this is a primary active transporter that uses ATP, and it uses ATP to move calcium in which direction?
Out of the cell.
Absolutely. It is going to use ATP to move calcium out of the cell. Excellent. So it is going to directly use ATP to move calcium out of the cell. Now, these voltage-gated calcium channels are open, and there's a massive amount of calcium entering into the cell. These pumps are going to be pumping it out. But if you're in a little rowboat and I drill 47 holes into the bottom of your rowboat, and then I give you a hand pump, that hand pump's going to help you pump water out of your boat. Are you going to be able to keep that boat floating?
No.
No, because you have a lot more calcium coming in than these poor little pumps can pump out. So yes, the pumps are there. Yes, they're trying to get rid of calcium, but far too much calcium is coming inside. And so since so much calcium is coming inside, we get an increase in calcium levels in the cytosol. And as we know, calcium makes cells do wonky things. In this case, calcium, and watch the wording here, triggers the release of acetylcholine via exocytosis. Calcium doesn't come in the cell, grab the vesicle, bring the vesicle to the plasma membrane, cause that vesicle to fuse with the plasma membrane and release its contents. It doesn't do it by itself. In fact, there are 25 proteins involved in this process of bringing the vesicle to the plasma membrane and causing it to be released. But calcium triggers the process. So calcium's not grabbing the rope and bringing it to the plasma membrane itself, but calcium triggers the process and causes our acetylcholine into the synaptic cleft. All right. So far, so good?
Yep.
Now, let's go back a step. As I mentioned, You have to be better about reading the questions. You have to be better about answering all parts of the questions. But the other thing that people need to worry about getting better at is being descriptive in their essay questions. It is 100% accurate to say that communication at the neuromuscular junction starts with us making the decision to produce an action potential, which causes acetylcholine to be released into the synaptic cleft. That statement is 100% correct. But what you're lacking is all of the details. One of the important things to think about when you're answering this, the essay questions, is what is the immediate effect of what you just described. Let's go through it again. We make the decision to contract a muscle. That produces a neural action potential. That neural action potential spreads down the axon to the synaptic end bulb. That neural action potential depolarizes the synaptic end bulb to threshold. That opens our voltage-gated voltage-gated calcium channels. Calcium enters into the cell. Calcium triggers the proteins to take the vesicles of acetylcholine, bring it to the plasma membrane, and release them into the synaptic cleft via exocytosis. Notice both of those descriptions were 100% correct, but one had much more detail, so one is going to get a whole lot more points. Make sure you are describing the processes, right? Don't just say that calcium enters a cell. Say that it is a voltage-gated calcium channel, right? Don't just say that the neural action potential causes the channels to open. Say that it depolarizes the cell to threshold, and that opens the voltage-gated calcium channels, right? Make sure you are being specific. Make sure you are describing in detail. That is what I'm looking for. That is how you maximize your points. All right. Now, we're still not done. We've released our acetylcholine into the synaptic cleft. And is it just going to stay there huddled, scared along the side of the synaptic end bulb?
No.
No, of course not. Our acetylcholine diffuses across the synaptic cleft. And reaches the motor end plate. Now, again, as we've talked about, this acetylcholine esterase is going to be finding and gobbling up some acetylcholine, but we're releasing buckets full of it. Are these acetylcholine esterases going to be able to gobble up all of the acetylcholine? No. So plenty of it is going to be able to reach our chemically gated cation channels. And that's what happens. Our acetylcholine binds to the chemically gated cation channel. And when it binds to that chemically gated cation channel, ions are going to move. Cations are going to move. And what are the two cations that we said are going to move?
Sodium, potassium.
Excellent. Which way is sodium going to move?
Into the muscle cell.
Yeah. Into the cell. Excellent. And which way does potassium move?
Out of the muscle cell.
So when these channels open, we are going to get the way too big. The two-way movement of materials. Sodium is going to enter into the cell, and potassium is going to exit the cell. Now, if one positive sodium comes in and one positive potassium leaves, is that going to change my muscle cell at all?
No.
No. But it turns out the movement of these materials are not even. As it turns out, there is a lot of sodium that enters. So actually lots of sodium enters the cell and only a little potassium actually leaves. So it's not an even movement of ions. A lot more sodium enters the cell and only a little bit of potassium leaves. So what is the net effect of that going to be? What's going to happen to my muscle cell?
Depolarizes the cell.
Excellent. Perfect. Our muscle cell gets more positive. And you are absolutely correct. There is a fancy word that we can use for that. The muscle cell depolarizes. And if the depolarization is enough, and how would we know if the depolarization is enough? Wouldn't there be some critical membrane potential we would need to get to?
Yes.
-6.
Yep. And what would we call that?
Threshold.
There you go. If its depolarization is big enough, oops, let's say it that way, to reach threshold, which as you pointed out, we know is -60 millivolts. If it's big enough to reach threshold, then the muscle cell produces a muscle action potential at the motor end plate. And once again, if we produce a big, huge wave, is it going to stay just here at the motor end plate?
No.
No. So our muscle action potential spreads down the sarcolemma. So we get that huge, big positive signal here at the motor end plate, our muscle action potential, and that muscle action potential will then spread down the sarcolemma. And there you go. Just that simply, we have initiated communication at our neuromuscular junction. We have accomplished our goal to convert a neural action potential into a muscle action potential. Questions on that? Stunned silences. Excellent. I love it. That means you all completely understand this. So let's take a look. Let's go through the process again. All right. Here, we're at our neuromuscular junction. Whoops. Oh, I thought I had all the pretty words here. I guess not. Okay, here we go. Starts with the decision. Neural action potential is produced in the brain. It's a big positive signal that depolarizes the cell. That signal travels down the axon, reaches the axon terminal, and reaches the synaptic end bulb. Again, we want to think of terms of the immediate effect. What is the immediate effect of it reaching the synaptic end bulb? It depolarizes the voltage-gated calcium channels to threshold. And when it reaches threshold, our voltage-gated calcium channels open. And when our voltage-gated calcium channels open, which way does calcium move?
Into the cell.
Yeah, calcium enters the cell. Calcium, remember, triggers the exocytosis. It doesn't actually grab the vesicle and bring it to the plasma membrane, but it triggers those 25 proteins to do their job of grabbing the vesicle and bringing it to the plasma membrane and releasing the acetylcholine from that vesicle. And that process we call exocytosis. Once in the synaptic cleft, it diffuses across the synaptic cleft and the acetylcholine finds those chemically gated or ligand gated. Remember, we can use those terms interchangeably. Cation channels. And those cation channels open. The two cations that primarily move through this cation channel is sodium and potassium. As we know, sodium is going to enter the cell. As we know, potassium is going to leave the cell. But as we talked about, do equal amounts of both move? No. Which one is there more of moving?
Sodium.
Right. So only a little bit of potassium leaves the cell, but a massive amount of sodium comes in. So we have a lot more of our positive ion entering the cell and very little of our positive ion leaving the cell. The net effect is our cell is depolarized. And remember, it's not hardwired. So if and only if that signal is big enough to reach that magical point again, threshold, then we will generate a muscle action potential. And that muscle action potential will spread down the axon. Questions on this? All righty. Stunned silence means we understand it perfectly. Dr. Slutsky, please continue. And in fact, go faster. So let's do that. We have accomplished our first goal. We have initiated communication at the neuromuscular junction. But remember, the same way we need to worry about turning the process on, and after all, that's what this is here, is turning the process on. We also have to worry about turning the process off. And turning the process off is what we call termination of communication at the neuromuscular junction. Let's move this down here so it's out of my way. I can move this up here and this here. That gives me more space to play with. Excellent. Oh no, hold on. All right. Get rid of that. This is the turn it off. And again, it doesn't hurt at this point to think in terms of what our goal might be. for termination. Our goal for termination at this point is to stop producing muscle action potentials. After all, the muscle action potential was the goal of turning this on. So our goal of turning this off is going to be to stop producing muscle action potentials. And that'll help us to focus on the process that we need to worry about. All right. So let's add this on. And of course, where is this going to begin?
Brain.
Yeah. And what happens in the brain?
You make a decision.
Absolutely. It starts with, again, making the decision. In this case, the decision is I want to relax the muscle. When I make that decision that I don't want the muscle to contract anymore, basically I stop sending neural action potentials. Actually, let's. So I stop sending neural action potentials. So as a result of it, there are no new neural action potentials spreading down the axon. Now, as there's no new neural action potential spreading down the axon, remember a neural action potential made the cell more positive. But does a cell necessarily want to stay more positive?
No.
No, a cell wants to be at its resting membrane potential. So as soon as we stop depolarizing it, the neuron returns to its resting membrane potential. And of course, there's a fancy name for returning to its resting membrane potential. Anybody happen to know what that is?
Homeostasis?
True, it is the process of homeostasis. But remember that we gave a fancy word when a cell gets more positive, we say the cell depolarizes. When it goes back to rest, we give it a fancy term and we say it repolarizes. So the neuron repolarizes. Back to negative 70 millivolts. So no new action potential. Our axon repolarizes to negative 70. And guess what? Our synaptic M bulb repolarizes to negative 70 millivolts. What happens when our synaptic M bulb drops below negative 60 millivolts?
The voltage gated calcium channels close.
Exactly. So when our synaptic M bulb drops below threshold, our voltage gated calcium channels close. That means there's no new calcium entering the cell. All right, we comfortable with that?
Yeah.
However, we still have this issue where there's still a lot of calcium in the synaptic M bulb. So while no new calcium is entering, we have to get rid of the calcium inside, already inside, let's say it that way, the synaptic M bulb. And is there something that might allow us to do that?
Calcium pumps.
Yeah. Notice those calcium pumps weren't able to do a whole heck of a lot when all the calcium was rushing in. But now that there is no new calcium rushing in, our calcium pumps are able to remove the calcium from the cytosol. So our calcium levels drop. And when our calcium levels drop, there is no new exocytosis of acetylcholine. We stop releasing those vesicles. We've got no calcium, no new exocytosis of more vesicles. No more vesicles bind to the plasma membrane. No more exocytosis takes place. So notice once again, no new acetylcholine into the synaptic cleft. But notice once again, that's only half of our problem, because we still have to get rid of the acetylcholine that is already in the synaptic cleft. So yes, no new acetylcholine is being released into the synaptic cleft, but there's already a bunch of acetylcholine in here. So we need a way to get rid of it. And luckily, as it turns out, there are 3 ways that we can do that. What hopefully is the most obvious?
Acetylcholinesterase.
Absolutely. Pac-Man here is going to be gobbling it up. Oh, hold on. Let's cheat. Do that. Perfect. Acetylcholinesterase, ACHE, breaks down the acetylcholine. So obviously that is going to be a big, huge chunk of it. But it turns out it's not the only way. While it is the biggest, it's not the only one we rely on. As it turns out, some of the acetylcholine will be reabsorbed by the synaptic end bulb. It'll actually be taken, bundled up into a vesicle, and brought back into the synaptic end bulb. And did we have a fancy name for that type of process?
Endocytosis.
There you go. So we'll have that endocytosis of the synaptic end bulb. And notice one other thing. While our 2 cells are very, very close to each other, they don't touch. And that synaptic cleft has the nooks and crannies to try to keep anything in here. But are there walls on the outside of that?
No.
No. Is there anything stopping an acetylcholine from just floating away?
No.
No. And so the third thing that will happen is some of the acetylcholine diffuses out of the synaptic cleft. Right. So again, some of it will be bound back into a vesicle, brought back into the cell via endocytosis. But there's nothing stopping one of these from floating away. And notice, this acetylcholine over here next to the muscle cell, can it have any effect?
No.
No, because all of the channels are back here in the motor end plate. So I could dump a whole bucket of acetylcholine over here on this side of the muscle cell, and it's not going to have any effect because it's going to be away from the channels. So endocytosis, Pac-Man, and diffusion. Those are the 3 ways we can remove all the acetylcholine from the synaptic cleft. And of course, once all the acetylcholine is gone from the synaptic cleft, so that's gone, and that's gone, and this is gone, and that's gone. What's going to happen then?
The ligand gates close up.
Exactly. With no acetylcholine, then with no acetylcholine, and let's write it down here. Our chemically gated cation channel close. Of course, that means no new potassium out of the cell, but was potassium really driving the process? No. So no new sodium into the cell. As we know, cells have pumps, etc., to get back to resting membrane potential. And what did we call that process of getting back to resting membrane potential again?
Repolarizing.
Repolarization. And when our motor end plate repolarizes, no muscle action potentials are produced. And lo and behold, we have accomplished our goal. No new muscle action potential, no muscle action potential spreading down the sarcolemma. There we go. Questions on that?
Yeah, just one. Is it just the absence of calcium in the synaptic end bulb that causes the acetylcholine to go back into the bulb?
No, endocytosis will be occurring spontaneously, but again, more acetylcholine is going to be coming out than will be coming in. And so the same way we were talking about calcium, those calcium pumps are always active. But if more calcium is coming in, then it has a negligible effect. And it's the same thing with the endocytosis. Endocytosis may be occurring, but exocytosis is happening much more rapidly. So there's far more acetylcholine leaving the cell than there is coming in. So it's, again, it's only when there's no new acetylcholine being released that that endocytosis really has an effect. Yep.
A question too.
Yep.
During the beginning process of termination, are more acetylcholine esterase being released, or is it just the fact that everything is being turned off and no new chemicals are being produced, proteins?
Correct. Yes. There is always about the same amount of acetylcholine esterase in the synaptic cleft. So again, it's a numbers game. I always think of it. I always think of it in terms of like if you've ever seen those nature videos of the baby, the hatching of baby turtles, right? The moms lay their eggs on the beach, and hundreds of these baby turtles get out of their shells, and they have to make that treacherous, crawl back to the water so that they can survive. And the whole time they're doing that, all these seagulls are coming down and grabbing baby turtles and eating them, right? But as long as there are more baby turtles going towards the water than there are seagulls to eat them, some of the turtles make it to the water and the species gets to continue to survive. And it's the same thing here. More acetylcholine is being released than can be gobbled up by the acetylcholine esterase. So it isn't until we stop releasing it that it can have a significant effect on decreasing the amount that's in there. Because as much as they can gobble up, more is being released by the synaptic end bulb, so we get an effect on the motor end plate. Yeah, and that's a great point. All of these things, here, I'll do it this way. The calcium pumps, the endocytosis, the acetylcholine esterase, these are processes that are occurring all the time. But notice they're only having a significant effect during termination. Because during initiation, more calcium is coming in that can be pumped out. During initiation, more acetylcholine is being released than can be gobbled up by the acetylcholine esterase or can be endocytosed by the synaptic envelope. So more comes out and we have an effect. More calcium comes in and we have an effect. So yeah, these things happen all the time, but they're only having an effect during termination because that's when we're stopping more from coming in or going out. All right. Excellent. Let's look at the pretty pictures from your textbook from this as well. Like we said, we want to first stop removing acetylcholine. So we make the decision. We repolarize the cell. We stop releasing acetylcholine from the synaptic envelope. But remember, that doesn't solve all of our problems. We still have to get rid of the acetylcholine that's in there. Some of it diffuses away. Most of it is broken down by acetylcholine esterase, and a little of it is endocytosed. We get rid of the acetylcholine, sodium stops entering the cell, and our muscle action potential stops. All right. So really the key to terminating the muscle is getting rid of this acetylcholine, but obviously there's a big long process in that. All right. Questions on this?
Nope.
All right. So as I mentioned, if we truly understand a process, then one of the ways we can show that understanding is by messing it up, changing something, and then using our knowledge of it to understand how that would affect it. Let's talk about a fairly familiar example of this. Botulinum toxin. Botulinum toxin comes from what?
Botulism?
Botulism, absolutely. And where do you typically find botulism?
Pig disease? Does that have to do something with pigs?
Not necessarily, although that's a good guess.
Is it canned goods?
Yeah, typically canned or jarred goods that aren't canned or jarred properly, right? You go down into your storage, facility downstairs and you bring up that can of beans that is absolutely bulging at the seams. Is that the kind of thing you want to bust open and start eating right away? No, because typically botulism has gotten in there and it produces a very deadly toxin. Let's talk about what botulinum toxin actually does. So again, our goal now is to get an understanding of our modification of components process of communication at the neuromuscular junction. And as I mentioned, the first example of this that we're going to look at is the botulinum toxin. With the botulinum toxin, what it actually does is, remember how I mentioned that there are 25 proteins involved in the process of bringing that vesicle to the plasma membrane and releasing it via exocytosis?
Yeah.
All botulinum toxin does is break one of those 25 proteins. It happens to be a protein called SNAP, but you don't need to know that. But when it breaks one of those 25 proteins, what happens is it stops the process of exocytosis. So let's think about what that would do. Of course, we start with the decision, check. We produce a neural action potential, check, which spreads down the axon, comes to the synaptic M bulb, opens our voltage gated calcium channels. Calcium comes rushing into the cell. And then what happens?
The toxin breaks the 25 and therefore it can't be released. And now the cell does wonky, wonky things.
Exactly. And in this case, the wonky thing is the cell, or let's be more specific, the synaptic M bulb cannot release acetylcholine. All right. Because of that damage, our synaptic M bulb cannot release acetylcholine. That means no acetylcholine into the synaptic cleft. So what happens to our motor end plate?
Doesn't get stimulated, doesn't open up.
Yep, means no sodium entering the cell. And if no sodium enters the cell, does not depolarize. And if it doesn't depolarize, what happens?
No muscle action potential.
No muscle action potential. As a result of that, our muscle cannot contract. I can send a signal as much as I want to that finger to try to get it to move. But no matter how many signals I send, it's not going to be able to contract. It produces what we call a flaccid paralysis. Right? Most of you are sitting in chairs right now. And if you reached down and poked your legs, you would say the muscles in your legs are relaxed. However, if I wheeled a paraplegic into your room and you poked his legs, would his legs feel the same as your legs?
No.
No. While yours are at rest, remember we've also talked about how muscles have muscle tone. That helps to support the joints, helps to protect the organs, helps in circulation, all sorts of other things. That because you're sending basically partial signals to your muscles so that they're in a partial state of contraction and a partial state of relaxation. That paraplegic, because of the damage to their spinal cord, no signals are going to their legs and their muscles are completely flaccid. And that would be what your muscles would be like in that situation as well. Now, as I mentioned, a common place you find botulinum toxin is in spoiled, canned, and jarred goods. But as we've also talked about in this class, once we understand how something works, we can use it to our advantage. And where's the other big location where you find botulinum toxin these days?
Botox.
There you go, plastic surgeons' offices. Of course, if you use a gross name like botulinum toxin, nobody wants to stick that into their skulls. But if you give it a cutesy name like Botox, then sure, we'll have Botox parties. Let's all get together and drink wine and get drunk and inject each other in the forehead with botulinum toxin. Why do we put that Botox in our forehead?
Relaxes the muscles.
Exactly, right? You have these pesky things that happen when we get older called wrinkles because our skin loses some of its elasticity. And as I just finished mentioning, our muscles have some tone in them. And so as our skin loses their elasticity, right, and our muscle has that tone in it, it causes these pesky wrinkles to appear. But if I paralyze the muscles by damaging the neuron so that it can't release acetylcholine, then suddenly my forehead is completely smooth and flat. Yes, I can no longer show emotions, but as long as my forehead is smooth and flat, that's all that matters. Because as we know, looking good is the only thing that matters in life. All right. Now, of course, I jest, and obviously there's a tremendous vanity to it, but that doesn't mean that there isn't some good stuff that botulinum has been able to help us with. It can be used to help in some types of migraines. Some migraines are actually caused by a constriction of superficial blood vessels of our scalp. So what they will actually do is they will go in and inject botulinum toxin into those so that those smooth muscles dilate, no longer can constrict, and that can stop the migraines from occurring. People who are produce excessive amounts of sweat, they will inject this into the sweat glands, because the same process by which our neurons release acetylcholine is the same process by which sweat is released using the merocrine mode of secretion via exocytosis. So we can stop the release of sweat in those regions with the botulinum toxin. But this is damaging the neuron, right? Luckily, our cells can repair themselves. Of course, first our neuron has to realize that that protein has been damaged. So it has to send a signal all the way back up that axon back to the cell body to tell the cell body to make new proteins to replace the ones that are damaged. Then it has to bundle those up in vesicles and send it down the axon back to the synaptic end bulb. And that process can take weeks to months, right? When grandma gets that Botox in her forehead, is her forehead smooth forever? No, eventually in months, the tone comes back and she has to do it again. And that's the problem, right? This Botox can get into the bloodstream and move away from the forehead to other muscles in your body. Like, oh, I don't know, how about your diaphragm? Would you necessarily want to damage the nerve that is releasing acetylcholine to telling your diaphragm to contract?
No.
No, absolutely not, right? You'd have a whole heck of a lot of problems breathing. The good news is that in a couple months, that protein can be repaired, but how long can you hold your breath for? Probably
Couple
not
months.
months. Yeah, exactly. So again, as we understand how this works, we use these things to our advantage. And so here we see how this modifies communication, the effect that flaccid paralysis, and then we can talk about what it does as well. Let's talk about another example. Another toxin that can modify communication at the neuromuscular junction is curare. Anyone know where curare comes from? There are a fair amount of mushrooms that produce curare. There are some like poisonous frogs and things like that that can produce a curare-like substance. Again, you may not be as familiar with curare as you are with botulinum toxin, but anyone who's ever watched an adventure film like Indiana Jones or something like that has some familiarity with curare. Curare is typically what is seen in that chemical that is on those poisonous darts that the indigenous people use. So you see our protagonist, like Indiana Jones and all of his colleagues, fleeing from the indigenous people, and the indigenous people are using those blow darts. And of course, all of his compatriots all get hit and collapse and fall to the ground. But of course, our hero gets away with whatever the prize is at the end. Well, curare is another toxin that also causes a flaccid paralysis. But it does it in a much different way. What curare actually does is binds and blocks the chemically gated cation channel. Right? Back in ancient times, and by ancient times, I mean when I was in high school, there was one class that I was taking, an English class, where we had a big, huge test. And one of the students who was not successful in the class thought that his way of getting out of the exam would be to take gum and put gum inside of the lock for the door. Because if the teacher couldn't unlock the door and get us into the classroom, then we wouldn't have to take the test. And so that's what he did. Not, of course, realizing that there are 2 doors into the classroom, one from the inside and one from the outside of the building. And so we were still able to get in, and it didn't stop anything, but he thought he was clever. And that's exactly what he did. He took his gum and he put it into the lock. And that's what curare does. Curare binds to and blocks the receptors of our cation channels, chemically gated cation channels. So again, we go through the process. We make the decision, check. We produce a neural action potential, check. It spreads down the axon. It depolarizes the cell. It allows calcium to come in. Exocytosis takes place. But now with all that acetylcholine in here, can it bind to the cation channel and have any kind of effect?
No.
No. You could dump a whole bucket of acetylcholine into the synaptic cleft and nothing is going to happen. So it binds and blocks the channel. So once again, sodium cannot enter. And if sodium cannot enter, no muscle action potential. All right. And again, like I said, many indigenous tribes use these type of chemicals in things like poisonous darts and things like that, causing the muscle to fail, causing people to fall down and stuff along those lines. But once again, our technology, our understanding of this allows us to use this to our advantage. Notice one of the big differences about curare is curare really isn't damaging either of the cells. It's not damaging the neuron. It's also not really damaging the muscle cell. Yeah, it's gumming up the channel, but it isn't breaking a protein the same way that botulinum toxin does. And again, this is something we can use to our advantage. One of the things that you're seeing more and more of these days are these incredibly elaborate transplant surgeries. People are getting hands transplanted. People are getting faces transplanted, right? You get mauled by a pet monkey and we can actually put a new face on top of you, the one that the monkey removed. Now, obviously, putting a face onto somebody is an incredibly delicate procedure. You have to connect all the blood vessels. You have to connect all the nerves, all these tiny little muscles to the skin. And as we just finished talking about, we know muscle has tone. Our muscles are undergoing tiny little contractions that are producing a tiny amount of tension that helps to hold our joints in place and things along those lines. And that's great for maintaining posture or while your leg is relaxed. But if you're trying to connect tiny little blood vessels and nerves on a portion of skin, do you want any even tiny muscle contractions taking place in that region? No. So what they will do is they will bathe the portion that they're working on with curare, causing a completely flaccid paralysis in that one region. They can then make all the surgical connections that they need, and then they just flush the interstitial fluid. And when they flush that interstitial fluid, that flushes the curare away, and then it goes back to its normal function that it was doing before. So again, the more we understand this, the more we can use this to our advantage. And let's talk one more example.
Question for
Yeah.
Professor. With curare, is that just like a localized thing?
Yeah,
Or
they're
will
just putting
that
a
spread out through?
because it is just in the interstitial fluid, they will put it local. Some diffusion may occur, but if you think about it, if a little diffuses to another synapse in another location where it only blocks 1 or 2 channels and not all of them, then it's not going to affect the function too much. It's only in that one area where they've got a concentrated amount where they're able to paralyze it immediately. And then when they're able to then, because again, remember, this is an open surgery, they then flush that interstitial fluid out of there. And then basically it's gone, and the neuron go and the muscle go back to its normal function.
Okay, yeah, I was talking more like in the real world, like if you come across it on one of those toads or frogs or something like that,
Or you get hit by a blow dart.
or you get hit by a blow dart, does it spread through?
Well, so again, yes, in the area, so they hit you in the leg with that blow dart, and it paralyzes the muscle where there's the highest concentration. But as it gets into the blood and diffuses out, the more diffuse that chemical goes, the less of an effect that it's going to have. Yeah. Is it possible if you ingest enough of it, for instance, so like you eat those mushrooms instead of having a dart or something like that, could the amounts of it be high enough where it would paralyze your respiratory muscles or something like that? Yeah, absolutely. Yeah. Which is why you shouldn't eat random mushrooms you run into in the wild. Right? Buy your mushrooms at Safeway, just like God intended. All right. All right. Questions on that? Excellent. One more example. Tetanus. Where do you get tetanus from?
Rust.
Rusty metal, absolutely. Step on that rusty nail or something along those lines, and the tetanus toxin can get into your body. All right, let's talk about what that tetanus toxin does. And I think I have one more color of green here. So let's see what we got here. Perfect. We'll use this one. So looks way similar to the previous one. There we go. I think that works. So tetanus toxin. Tetanus toxin, like curare, binds to the cation channel. Actually, let's cheat. I will move this down here so that we have a little bit more room. I guess I got to make it smaller to make sure it all fits. Tetanus, like curare, binds to the cation channel, but unlike curare, when it binds, it opens the channel. So what happens here is our curare chemical binds to the channel and doesn't let go. And this channel stays constantly open. And if this channel is constantly open, what's going to happen as a result of that?
A lot of sodium coming in and keeps it flexed.
Exactly. We are going to get a massive amount of sodium. Sodium enters the cell and doesn't stop. As a result of that sodium continuously entering the cell, our cell is in a constant. I have to put it over here because I'm running out of room. Let's say it this way. Let's go through all the steps. Cell constantly produces muscle action potentials, and so it is in a constant state of contraction. We call this a rigid paralysis, where the muscle contracts and doesn't stop contracting. In fact, what is the common term that they use for someone who has tetanus poisoning?
Lockjaw.
Lockjaw, exactly. Because basically the muscles of the cheek contract and continue to contract and don't let go. And so as a result, you're not able to open your muscle. If you look at some of the early medical journals where they talk about this type of condition of lockjaw, the illustrations they have are truly amazing, of these people who are just contorted in these horrible positions from this constant contraction of the muscles that will not stop. It is an incredibly painful process that can occur. And when it occurs in large parts of the body, it can be very damaging. These muscles are contracting at their maximal rate continuously, damaging the organs, damaging the bones, damaging the, the muscles themselves. It is an incredibly, incredibly damaging and exhausting process. All right. And if you think about it, as we talked about, with a flaccid paralysis, if all my muscles are loose, ventilating me is pretty easy. If all of my respiratory muscles are in a constant state of contraction, is it going to be easy to get air into and out of my lungs? No. So it's also a very, very dangerous condition as well. So there you go. We have now truly accomplished our goal. We have initiated communication at the neuromuscular junction. We have terminated communication at the neuromuscular junction, and we have now modified communication at the neuromuscular junction. We have mastered the process of part 1 in our muscle contraction. Communication at the neuromuscular junction. All right. Questions on that? I know this is a lot, but remember, as we have talked about, hopefully you are taking advantage of the resources. I cannot emphasize enough how much I like these A&P flicks that we have here in your study area. As your book's a great resource, does a great job of showing these things. We have lots of other great resources that show these things. But especially if you're like me, if you're a visual learner, going to these animations and videos, going to the A&P flicks. We watched this last time, but we'll throw it up again this time. Again, now that we've gone over it.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of the skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across a synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways.
3.
1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholine esterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
But remember also, some of the acetylcholine whole will be taken back up into it via endocytosis as well. So don't forget that third way as well. So there you go. As I mentioned, hopefully you've looked at that already. If not, you've got that tool to help you as we continue to go through that. All right. So we're 1/3 of the way through the process of contracting a muscle. The good news is steps 2 and 3 are not nearly as elaborate. Step 1 is definitely the more elaborate process. But the good news is that means we've gotten the hard part out of the way. That is all that I wanted to do for today in lecture. So I'll ask the question one more time. Any questions on these processes? I know as we look at the whiteboard, I have absolutely made an absolute mess of it. But you'll be happy to know that I also saved it all along the way as well. So I have both just the anatomy, just initiation, termination, and modification. I have those 4 pictures and I'll post them all onto our Canvas site. But obviously, I don't know, I worry that they won't make a lot of sense by themselves. So make sure you understand these, look at the lecture, read the textbook, look at the other resources to help you to be comfortable with this. So any questions on any of the 3 processes? And again, remember, notice now we have 3 possible essay questions, and I've guaranteed you at least 2 essay questions on this will be present. There's going to be a total of 7, and I guarantee 2 of them will be on the exam. So this is 3. We're almost halfway through those. So one of these is almost certainly going to be on the exam. All right. Any questions on that before we finish with the lecture and move to the lab? All right, excellent. Then let's go ahead and take our last break. We'll go ahead and get rid of that. During this break, make sure to grab, so let's actually do this. Actually, I'll share this as a reminder. So this will be our starting point. Remember, we're going to be talking about this. We will restart in 15 minutes. It looks like 10:47. I'll start the recording at that point. And remember also to grab a red and blue pen or pencil, plus that handout. So again, you've got that origins and insertion handout. And actually, let's leave it on that. There you go, this one. So grab this handout, grab a red pen, grab a blue pen or pencil. Pencil might be better because you can erase it. And we will restart at 10:47. All right, any questions? All right, I will see you guys in 15 minutes. We have now switching gears into lab mode. And as I mentioned, and as you've seen on your handout, you have a list of muscles that you're responsible for and all of their actions. However, also for the bolded muscles, you are responsible for the origins and the assertions. These are basically the attachment points for the muscles. And there are, of course, rules on what is what. Obviously, let's do this simply. A muscle almost certainly needs to cross at least one joint, because after all, if you had a muscle that just went from one part of one muscle to another part in another, when you contracted that muscle, it wouldn't really do anything. So instead, a muscle has to cross at least one joint. And so typically we'll have 2 attachment points. Now, the primary way we move, we use the definitions of what is an origin and what is an insertion is the way they move. If you think about it, if you have a muscle across two bones, when you contract that muscle, the two bones don't come up and meet in the middle. Typically what happens is one bone stays stationary, and then when you contract the muscle, the other one moves. So typically when a muscle contracts, one of these bones will move and come up. Whereas the other stays stationary. So the stationary end is typically what we would call the origin. And the movable end is the one that we call the insertion. So when we talk about the origin and the insertion, what we can say is that when a muscle contracts, it pulls the insertion towards the origin. All right, so that is basically the action of a muscle. When it contracts, it pulls the insertion towards the origin. And like in this case, it can change the angle of the bones or have any other normal effects as well. And remember, when we talk about the actions, we can do that two ways. We can talk about the joint it affects, joint or joints, or we can talk about the part of the body that moves. So again, this action right here, we could describe two ways. We could describe it as a flexion of the elbow. That is the joint where it is taking place. Or we can say, I flexed my forearm. That is the part of the body that moves in space. So when we are talking about the actions, when we're talking about the origins and insertions, these are the things we are talking about. Our fixed end is the origin. Our movable end is the insertion. Now, is this true 100% of the time?
No.
Yes and no. This is the definition of what we use for an origin and insertion, but as we know, the sky is blue. There are going to be some exceptions. We'll see some examples of this. A great example of this involves some of the muscles involving my head. With this, I can bring the insertion towards the origin, and I can flex my head, for instance. But if I use other muscles to stabilize my head in place, when I contract this muscle, instead of my head going down, my ribs can go up. So some can be reversible. So you can put a little asterisk that way. So we want to do that because it can be, especially like in the legs and muscles that cross multiple joints, you can see that as well. So we'll see some examples of where this can be reversible. But there are also some other characteristics that tend to be true. Again, key word here being typically, right? That means that there are exceptions, but typically the origins are either more proximal or closer to the midline of the body. Typically, origins have a broader attachment point, so they're typically wider in their attachment, whereas insertions are more narrow. But here's the other big key important thing we have to remember. Here in my arm, we have a superficial muscle that most of us know the name of. What is that superficial muscle again?
Bicep brachia.
Biceps brachia. Brachia, of course, refers to the region of the body that it's in. Biceps, of course, refers to the fact that it has two origins. There are some muscles that have two or more heads, or we also sometimes use the term body. Those can be interchangeable in them. And each one has its own origin. So on the exam, for instance, you don't need to just know the origin of the bicep brachia, but as we talked about the biceps brachia, has two heads, and each of those two heads, oops, and each has its own origin. And you are going to be responsible for each one individually. On the exam, for instance, you may see a model or a chart or something, and let's say this is an arm, and there's belly 1 and there's belly 2, and they come together into a single insertion, and they each have their own origin. On the exam, I may have an arrow pointing to this one right here. And when I do that, there are two specific questions I can ask. The first one is to identify the muscle. And the second one will be to identify the origin. Now, notice if I have a second arrow, I'll make the second arrow green, and I do that here. Will the answer to these two questions be the same or be different for those two arrows?
Different.
They're going to be different. And so there will be a magical term I will use on the exam, which is a dead giveaway that I am looking for a very precise piece of information. And that very specific term will be specific. So, for instance, one question could be identify the muscle, be specific. And in this case, if it was the bicep brachia, does anybody know the two names of the bicep brachia heads?
Short head and long head.
Short and long, absolutely. So for that, your answer, if it was the red arrow, would be the long head. If it was the green arrow, would be the short head. And if you see that magical term, be specific, then make sure you are specific. Don't just say that is the short head and the long head of the bicep brachia, because that is not specific. In fact, if you put that, you will not get credit for that. You can't put both and just hope for the best. If you see that magical term, be specific, you need to be very precise with your information. And if you're not very precise, you will not get credit for it. All right, same thing with the origins. I can say identify the origin of the muscle, be specific. And if I do that and you write down the origin of the short head of the bicep brachia is the coracoid process of the scapula, and the origin of the long head of the bicep brachia is the supraglenoid tubercle of the scapula, while that information is 100% correct, you will get that wrong on the exam. Because I didn't ask you for both, I asked you for one specific one. So make sure you identify which specific one it is and just give me the specific answer for that. It's the only time you'll see this term be specific. So if you see the term be specific, that is a dead giveaway that it is a multi-headed muscle. And I'm either asking you for the precise name of that belly, that head, or the precise origin of that. Because notice, it only has one insertion. So the insertion is going to be the same for both, and the actions are going to be the same for both. So if you see that magical term, be specific, I'm looking for a specific muscle or a specific origin. And so make sure you are specific that way. All right, questions on that? And again, we'll be going over these together, so I will remind you of that as we go through them. The other important vocabulary to understand is the term muscle group. Guess how we define a muscle group?
Muscles that are grouped together that cause a certain reaction or a certain movement.
Excellent. Okay, sort of. Absolutely. I like where you're going with that. Obviously, the obvious part of a muscle group is it's going to be two or more muscles, right? Because you can't have a group of one. Now, you are correct in that they will be similar in function, but notice they don't have to be identical in function. However, the key to a muscle group is that a muscle group shares one or more attachment point. So, for instance, your quadricep femoris is a muscle group that all share the same insertion. They all have the same insertion. Whereas your hamstring on your back of your leg are three muscles that all share the same origin. So they must have either one or more attachment points. So either the origin or the insertion. And some will actually have both. Now, if you look at your handout for this exam, there are some muscle groups that you only need to know the name of the muscle group. For instance, if you look at your handout on, I think it's page 1, where you've got the head muscles, one of the head muscles you have is known as the zygomaticus. The zygomaticus is a muscle group. What do you think the two muscles of the zygomaticus muscle group are?
Zygomatic bone.
Well, no, those are the origins and insertions, but it is a muscle group made out of two muscles. One is called the zygomaticus major, and the other is called the zygomaticus minor. Do you need to know the names of those two individual muscles?
No.
Yeah.
No, they're not on your list. Do you need to be able to identify each individual muscle?
No.
No, it's not on your list. I am just going to point at that collective muscle group and ask you to just know it as a single muscle. So that is a muscle group where you just need to know the name of the individual muscle group, not the name of the individual muscles. There will be some others, like your quadricep femoris on page 4. 1, 2, 3, 4 on page 4. The quadricep femoris is a muscle group you need to know. But do you need to know the names of the individual muscles? What do you think the names of the individual muscles that make up the quadricep femoris are?
Rectus femoris?
That's one of them. What else?
Vastus intermedius.
And?
Vastus lateralis.
And?
Vastus medialis.
There you go. So that is a muscle group, which you definitely need to know them collectively as a muscle group and know that name. But there you need to know the individual muscles. Zygomaticus is a great example of a muscle group where we're not going to worry about the individual muscles. So some muscle groups you'll just need to know as a muscle group. Some you'll know them as a muscle group, but you also need to know the individual muscles. And it's all right there on your list. Notice zygomaticus major and zygomaticus minor are not on there because you don't need to know them individually. You just need to know them collectively as the muscle group that is the zygomaticus. So again, I've tried to make this as straightforward and simple for you as possible. All right, questions on that? All right. Lastly, as we talked about in bones, names matter, vocabulary matter, right? There's a difference between a tuberosity and a tubercle and a trochanter, right? Names meant something. And names mean something in our muscles as well. There are basically three main ways that muscles get their names. And again, typically, so there are going to be some exceptions. But the first is where it's located. So the biceps brachia is in the brachial region. The rectus femoris is in the femoral region. So one of the ways they get their names is their location, right? Guess what the flexor carpi radialis does?
Flexes the carpals.
Well, yeah, it flexes the carpals, it flexes. Guess what an elevator does?
Elevates.
Yeah, elevates. So one of the ways that it gets its name is by its actions. And the third way a muscle gets its name, oops, I guess it didn't have it here. So location, function, and the third way it gets its name is the shape of the muscle. And of course, can shape affect function?
Yeah.
Absolutely. So it is important for us to have a basic understanding of the shapes of our muscles. When we talk about the shapes of our muscles, we know now that our muscles are made up of fascicles. And inside of that fascicle are dozens of muscle cells that are all parallel to each other. Why is it important to have all the muscle cells in a fascicle parallel?
Coordination.
Yeah, coordination, absolutely. So if this muscle cell contracts or this muscle cell contracts, does my fascicle do the same thing or a different thing?
Same thing.
Same. Whether one of these muscle cells contract or all of them contract, I'm going to get the same action to my muscle. So again, this coordination affects in the strength, affects the activity of it, the coordination, and is important that way. So in a muscle, all the muscle cells are parallel within one fascicle. However, in a muscle, not all fascicles are parallel. So for instance, in a muscle, I may have 3 fascicles that are all arranged like this. And now, if muscle cells in this fascicle fire and muscle cells in this fascicle fire, are they doing the same thing or are they doing different things?
Different.
Different things, exactly. So when it comes to how shape affects function, it is all determined by the arrangement of the fascicles. So when we talk about the shapes, we talk about how the fascicles are arranged, and that is going to give us our functions. And as you can see, there are 4 major shapes, 4 major arrangements of our fascicles that will influence the movements of our body. Let's take a look at these. Starting first with a parallel muscle. Guess how the fascicles are arranged in a parallel muscles?
Parallel.
Parallel.
Exactly, fascicles are parallel. So in this one, and this will be easy for me to draw, fascicle 1, fascicle 2, fascicle 3. Notice that all the fascicles, all the muscle cells are all arranged parallel. This is actually the most common type. Oops. Most of the muscles in our body have this type of arrangement. And again, as we talked about, the advantage of this is precision. If any of the hundreds of muscle cells in this fires, all the actions are going to be the same. So this gives us nice precision of movement. Notice it also gives the muscle kind of a spindle shape where it has a thickened belly and then it tapers to the two ends where it forms its attachments. But there is something important, something interesting about the arrangement of these muscles. As we know, when a muscle cell contracts, what happens to it?
Shortens up.
It gets shorter. And when these fascicles get shorter, what actually happens is they also get thicker as a result of this as well. So when the muscle contracts, the muscle gets shorter, but the muscle also bulges out. Again, you may not have thought of it in these terms, but I know you're aware of this, because what is the number one most important reason why you contract your muscles?
Get the girls.
Exactly. Well, although, see, when you say it that way, it doesn't sound right. However, if we say you use it to woo women, then that sounds much more harmless. Exactly. We made the joke earlier in this class, how, you know, you do the curls for the girls so that when you're on campus and someone asks you directions, oh, the library, oh, it's over that way, right? You can do those curls for the girls. We get that bulging of the muscle. So it is a very common muscle arrangement, very common type of shape. And this is an activity that we are familiar with. All right. Questions on that? Now, it's obviously some advantages because we get the precision with this, but there's sometimes where we don't necessarily want that precision. Notice here we have what is called a convergent muscle. Here, the fascicles fan out in a fan-like fashion. So they basically have a single attachment point and then a big, huge, broad, flat, fan-like distribution. What's the advantage of this type of arrangement?
Covers more surface area.
True, it does definitely cover more surface area, so that could provide more protection, and that's definitely something that's going to be useful. But also, let's think about, like we were talking before, if I contract this fascicle versus contracting this fascicle, am I going to get the same movement or a different movement?
Different.
Different. So the advantage of this is flexibility of motion. This is going to give us a much wider range of motion from this type of muscle. So with this one, we get this fan shape with a single attachment point, much more flexibility of movement. Where's an example of a place where we have this type of convergent muscle? Again, an obvious one most people are familiar with.
Pectoralis majors.
Pectoralis major. Pectoralis major is this big, broad muscle here. And as we talked about, one of the primary ways you work that out is by doing the bench press. However, if you truly want to work your entire pectoralis major muscle, do you always do your bench press just from a flat horizontal bench?
No, incline, decline.
No, sometimes you're going to be inclined, sometimes you're going to be being declined, so you're pushing up and you're pushing down. So that way you're doing the entire range of motion that that muscle allows. Again, not surprising where this is located. Our shoulder, as we've talked about, is our most dexterous joint with the most range of motion. So it's not surprising we have this big convergent muscle helping to allow that flexibility of movement. Excellent. Now, I've used the example many times of this class of us playing tug of war. me on one side of the rope and all of you on the other side of the rope. And if we were to play tug of war, me on one side of the rope and you on the other side of the rope, who's going to win?
Us.
You guys, absolutely. But what if the rope's only 3 inches long? If I've got a good grip on that rope and you guys, all of you have all of 3 inches to be able to grab onto, what are your chances of winning then?
Not good.
Yeah, not as well. So for you guys to have that strength that you need, to produce that strength you need, you all have to be able to get a hand on the rope. And that's great here in the forearm, we have all the parallel muscles, or here in the chest, we have the convergent muscles, right? But unless you're Popeye, you probably don't have a hugely massive forearm. But you can have a tremendous amount of strength in your forearm. And the way we're able to do that is what are called pennate muscles. Pennate muscles, their job is to get as many fascicles as we can onto a tendon. So what I have here is my tendon. And my goal is to get as many fibers, as many fascicles, I should say, as possible onto that. And so typically the way that occurs is by having them arranged not parallel to the tendon. They are parallel to each other, but they're not parallel to the tendon. And so in this fashion, the advantage of this type of arrangement is I can get a large number of fascicles onto a single tendon in a small space. So again, the real huge advantage to this is I can get large strength with a smaller muscle. I get lots of fascicles in a small compartment and giving me a lot of strength. Now, it turns out there are actually 3 types of pennate muscles. The first one is the unipennate that we see here, 1 tendon and 1 row of fascicles. And again, the muscles in a forearm are a great example of where we find these. But we can also have muscles that have 1 tendon and 2 rows of fascicles connecting to them. This is what we would call a bipennate. Best example of this one is actually your rectus femoris, right? Your leg is a big, huge, massive chunk of meat, right? If you want to quickly lose 20 pounds, cut off one of your legs. They're these big, huge, massive chunks of meat that we need to swing around space. So we need big, powerful muscles to do that. And right on top, right at the top part of your leg is a short, straight muscle that has 2 rows of fascicles. So it takes up a relatively small compartment on the leg. But by having this massive number of fascicles, it's one of the strongest muscles in our body. And I think I've got a pretty picture of this one as well. There we go. Notice a single tendon, 2 rows of fascicles going into it, and that is that long, straight rectus femoris muscles. However, also here on the lateral aspect of our shoulder, we need a powerful muscle to be able to move our arm through space, but it's not the 90s anymore, so we don't want to look like we've got these big, huge shoulder pads. So we need to have it in a relatively small space. And to do that, we have a multi-pennate muscle. Whoops, multi-pennate, that's not what I wanted. I wanted a box. With a multi-pennate, we have 2 or more tendons and obviously 2 or more rows of fascicles. This one is somewhat similar to a convergent, where we have lots of fascicles going in different locations, but it takes up a smaller space because we're able to, as you see in the cross section, we can wrap the fascicles around the tendons. So it allows, again, for more organization, a lot of power in a small space. But this one gives us a much larger range of motion. Whereas the unipennate and bipennate are basically just pulling in one direction. Now with multiple tendons, we can pull in multiple directions. So I can bring my arm up, I can bring my arm forward, I can bring my arm back. I have more range of motion thanks to that multipennate deltoid muscle. All right, questions on that? And that leaves us with the last arrangement of fascicles. We can have fascicles arranged in concentric circles. And not surprisingly, we call that a circular muscle. Typically, these form around openings because when you contract a circular muscle, what happens to the lumen in the center?
Gets smaller.
It gets smaller, right? So this allows us to regulate the size of our openings. Excellent, right? We can regulate the size of our openings that way. And in fact, in some areas, this muscle can thicken so much that when it contracts, not only does it decrease the size of the opening, but it actually closes the opening entirely. And if it closes the opening entirely, it can work as a valve. And what would we call the fancy name of a circular muscle that acts as a valve?
Sphincter.
There you go. So sphincters are basically enlargements of these circular muscle layers that regulate the flow of substances into, out of, and through our body. So again, lumen gets smaller and can even contract and form a sphincter. All right. Questions on that? All right. Excellent. With that, then, we are ready to tackle the muscles of our body. Now, as I mentioned, there are 600 named muscles on the body, on front and on back, and so on and so forth. You clearly don't need to know all 600 of them. You just need to know the ones on your list. And when you think of that in terms of your list and the fact that there's over 600 named, your list isn't that intimidating. But it still can be a little overwhelming. So much like we did in the bones of dividing it into axial and appendicular and skull and bony thorax and leg and all of that, we're going to do the same thing with the muscles. We are going to break them down into more easily digestible pieces. And the easiest way to do that is by the part of the body they're involved in moving. So let's start first with the muscles that move our head through space. Starting with a muscle that we already talked about, the masseter. The masseter is a superficial muscle located on the lateral aspects of our jaw. And as we mentioned, and the reason you've got your pens is we actually want to take the time, and let's make this a little bigger. There we go. You guys can see that okay, yes?
Yeah.
Excellent. Like I said, I guarantee on this exam, there will be skulls sitting there, pictures of skulls with arrows pointing at bone features. And you are going to be responsible for recognizing what muscle would attach to that attachment point, distinguishing origins and insertions. And as I mentioned, our rule by convention, and that is the rules that some anatomist made up somewhere, is that our origins are going to be red in color and our insertions by convention. And again, not my rule. If you look at any origin insertion images in your textbook, lab manual, on the almighty Google, you will see these rules as well. So let's start easy. What is the origin of our masseter?
Zygomatic arch.
Excellent. So, and again, you're not turning these in for a grade. So however you want to do this is up to you. The point of you doing this is to make it make sense to you. But as we know, there is my zygomatic arch made up of the zygomatic process of the temporal bone and the temporal process of the zygomatic bone. So I'm going to color that in so that I remember that. And what is the insertion?
Mandibular angle.
Excellent. So I can do that down here like this. Now, there are two reasons we are taking the time to draw these things. The first is because, as I've said, not only am I going to point to a picture of a masseter or a model of a masseter or something along those lines and ask you for the origin insertion, but I also will have skulls on this exam. We'll all point at the mandibular angle and ask you to identify the muscle that inserts into this bone feature, or I'll point an arrow at the zygomatic arch and ask you to identify the muscle that originates from this bone feature. But the other thing to remember is, as we know, muscles pull the insertions towards the origins. So the another advantage of taking the opportunity to look at these things is to realize that when this muscle contracts, it is going to pull the insertion towards the origin. And notice in that case, it's a pretty linear action, just one action. And what would we call that one action of the masseter?
Elevate mandible.
There you go. Elevate the jaw or elevate mandible. All right, brings it up. When would you use something like this?
Talking, chewing.
Talking, chewing, absolutely. All of those are great, great examples. However, again, you do want to be careful. Again, there are many possible answers, and most importantly, the correct answer is the one that helps you to get it right on the exam. However, for me, when I think of chewing, I always think of like that cow with its cud, where the jaw is kind of going all over the place. Notice this really does one thing. It just brings the jaw up. So this is like that first initial bite, right? So that's the one that helps me. If chewing helps you to remember that, all more power to you. For me, I think of that first initial bite, bringing that mandible up, elevating the mandible. That is the action of the masseter. All right. And notice you put your hands on your teeth, on your cheeks, and clench your teeth. You can actually feel that muscle contract on the side of your jaw. Now, let's compare this to another superficial muscle, the temporalis. What is the origin of the temporalis?
Temporal fossa.
Excellent. So we know here basically, oh, let's make that bigger. Otherwise I'll be coloring it forever. Here, that nice smooth portion of the temporal bone is where my origin is. Excellent. And I'll take the time to color it all in. And what's the insertion for this one?
Coronoid process of mandible.
Excellent. Now notice you can't fully see the coronoid process. So again, no wrong answer here. You're not turning this in for a grade. This is something you're doing to study. So do it the way you want to do it. If you just want to color the portion of it that you can see, perfect. If instead you want to do it really lightly, like as a dotted line or something, to show the part that you can't see, the part that's hidden by the zygomatic bone, you could do that. Again, there's no wrong answer. You just want to do something that makes sense to you. If you do what I just did there, and now suddenly you think the insertion's on the zygomatic bone, that's a problem. right? So make sure whatever you're doing makes sense to you. For me, I find it simpler to just put it on the part of the bone that I can see, and that's enough to help me to remember. And again, notice as we mentioned, when a muscle contracts, it pulls the insertion towards the origin. Notice this is a more angular motion. So unlike the masseter that just has one action, how many actions does our temporalis have?
2.
2. What are they?
Elevate and retract mandible.
Yeah, elevate, as we know, is to bring up. What does retract mean?
Bring in.
Well, to bring in, or retract means to
Bring back.
bring posteriorly, right? Move posteriorly. So it's moving posteriorly. So it brings the jaw up and brings the jaw back. Anybody think of something you would use this for?
The grinding the food between your teeth.
Yeah, obviously masseter plays an important role again in grinding and chewing. Again, if you close your mouth, put your hands on the side of your temples now, and you clench your teeth, you can feel your temporalis muscle, right? And in fact, if you lay your hands on the side, you can feel the masseter and the temporalis at the same time. However, again, I'm always thinking of activities that help me to remember precisely what the action is. And as I also often point out, the dumber something is, the easier it is to remember it. And of course, being an older sibling of 3 younger sisters, I am required by law to torture them as much as possible, make fun of them, especially when they say something stupid, right? And so how do you make fun of your sibling when they say something stupid? You make that durr face, right? That grrr, you're so stupid. Well, how do you do that? Elevate your mandible, bring it back to get the nice big overbite, and you do that nice big durr face, and you shame them into eating another Twinkie or whatever other psychological damage you're doing to them. All righty. This is how the game is played. Questions on this? Again, notice here we see these muscles. Again, masseter from the zygomatic arch down to the zygomatic angle. We can see that there. Here's our temporalis up here on the superficial part of the fossa. And obviously we can't see its insertion because its insertion is more deep, but we know it's connecting somewhere down here to that coronoid process. We can see that there. So we see the muscles, we know their origins, we know their insertions, we know their actions. And again, being the smart, sophisticated students that you are, if I have an arrow pointing somewhere towards the head region, and I ask you to identify the action of the muscle, or let's be more specific, the 2 actions of the muscle, which of these 2 muscles am I pointing at?
Temporalis.
Temporalis.
How do you know?
Because masseter only has one action.
Exactly. Conversely, if I'm asking for just one action and you're not 100% certain, which one am I pointing at?
Masseter.
The masseter, because how many actions does the masseter have?
1.
1. There you go. Again, I will be very precise in my wording of the questions. And as always, you can use that to your advantage. If I ask for the 2 actions of a muscle, guess how many actions that muscle has total?
2.
The 2.
2. Yeah, if I ask for just a single action, it only has 1 action. If I ask for 4 actions, that muscle has 4 actions. So I will ask you for the precise number of actions of that muscle. And so as a result of that, you can use that to your advantage on the exam. All right. Excellent. Let's move on to the next one. Again, there's always going to be these alphabet soup terms that you are guaranteed that you're going to have to spell at one point or another on the exam. And here's a great example of it. sternocleidomastoid. But like most alphabet soup terms we need to know, it tells you everything you need to know about it. Guess what sterno refers to?
Sternum.
Yeah, the sternum. Guess what clido refers to?
Clavicle.
Clavicle. And guess what mastoid refers to?
A mastoid process.
Mastoid process, exactly. There you go. This muscle has 3 attachment points. Now notice we can't quite see all of that with all of these pesky superficial muscles over the top. We see some of it, but conveniently enough for us, we can peel some of those away. And notice when we peel some of these away, we can actually see those specific attachment points. I'll draw them here and then we'll go to our skull in a minute. Notice clavicle, sternum, mastoid process. 3 attachments. Notice this isn't a continuous attachment. There are 2 separate, they're not quite bellies, but there are 2 separates. There's a little bit of a gap in between them. So let's go to our skull. Sternocleidomastoid. What is the origin of the sternocleidomastoid?
Manubrium of sternum and sternal end of clavicle.
Excellent. So over here on the manubrium, and then again with a little bit of a gap, the sternal end of the clavicle. Excellent. And there's a 3rd attachment point. That must be the insertion. And what is the insertion on?
Mastoid process.
Mastoid process. Now notice it doesn't show it on this image. Again, do what makes sense for you. If you wanted to cheat and put it over here on this one, where you can actually see the mastoid process and like draw an arrow, that would be fine. What I usually do is I just cheat. I know the mastoid process is sticking out back here. So I just go ahead and draw it on here. But if you want to give yourself a little arrow to remember, hey, it's actually this and inserts right there on that or something like that, if that doesn't get confusing. Again, this is your study guide. Do what makes sense to you. This is how I do it. But again, do what makes sense to you. All right. Questions on its origins or its insertions? Then let's talk actions. How many actions does this have?
3.
3. Now, the reason it has 3 is because how many sternocleidomastoid muscles do you actually have?
2.
2. 1 here on the left and 1 here on the right. And these muscles, not surprising, like many of the muscles that affect the median of your body, they can work together. In fact, if I contract both of these at the same time, it flexes my vertebral column, it flexes my head. In fact, the sternocleidomastoid is often sometimes referred to as the prayer muscle because it lowers the head in prayer. All right. However, remember, we also talked about how we can reverse origins and insertions. If I use other muscles to stabilize my head in place, when I contract my sternocleidomastoid, instead of my head coming down, I can bring my ribs, my sternum and my ribs up. But I also don't have to use both of them. Notice if I just use one, we know I pull the insertion towards the origin. So as I pull the insertion towards the origin, notice what it does is it rotates my head. So if I contract this one, if I contract the right one, it rotates my head that way. The left one, it rotates my head that way. So those are the 3 actions. One of them by themselves will rotate the head. 2 together can either bring the head down or bring the ribs up. Questions on the 3 actions? All right. Now, as I mentioned in the classroom, I actually break you guys up into groups and I give you these muscles and I make someone in your group demonstrate a common activity that is going to help you to remember this, right? Now, as I mentioned, I didn't do that for you guys, but I did encourage you, and you guys do have the handout. Did anybody come up with an activity that is going to help us to remember all 3 actions of the sternocleidomastoid? It's okay if you hadn't. We hadn't talked about any of these things yet. You might not have known what was expected of you. But I've had lots of great examples. A good example that I had in the classroom once was someone referred to, like that this is actually the one that you would use in church, right? Because again, not only is it going to bow your head, but also while you're sitting here, you may want to be gossiping about what, David up there is talking about. So you can turn your head side to side to be able to do that gossiping. And obviously you have to elevate your ribs to bring air in so that you can gossip, or if you're shocked by what David's wearing or something along those lines. I've had some really good ones, but my favorite actually happened about 10 years ago. Normally, somebody gives me something good. I watch another teacher and I learn something funny from them, or a student says something funny. Usually I give them credit for the first couple of years and then I take it as my own and take credit for it. But this I will never take credit for. It was awesome. I had this student, more than 10 years ago, sat in the back of the class, was very quiet, seemed very sincere, sweet, innocent, right? Just kind of sat back there chilling, doing her own thing. Kind of, like, I don't know, like Jacob, just sitting there looking sweet and innocent. But she got up and demonstrated what she referred to, her words. And again, remember, this was 10 years ago. It wasn't legal at the time. What she referred to as a bong rip, right? Flex the head. Elevate the rib cage and rotate your head. So there you go. You put those 3 actions together into a bong rip, and that helps you remember the 3 actions of the sternocleidomastoid. All right. Questions on that? There you go. Excellent.
Seems sincere.
I'm sorry?
I seem sincere.
Your name was the first one that jumped out at me when I looked over to the left where the list of the names were. So you're not sweet and innocent? You're telling us you're not sweet and innocent?
All in that.
All right, excellent. All right, excellent. So that is the sternocleidomastoid. Origin, insertion, actions. Questions on that? Here is one more view of the sternocleidomastoid. This one's from your textbook, and I really like it. Your textbook does a nice job of giving you all of this information. And again, we can really, really nicely, really well see it's attaching to the mastoid process up here on the skull, and then it has those 2 origins, one on the sternum and one on the clavicle. I make a point of emphasizing that because here on the other side, they demonstrate a muscle group for us. This muscle group happens to be the scalenes. The scalenes is made up of 3 individual muscles, the middle, the anterior, and the posterior scalenes. And are those names you need to know?
No.
Nope. You just need to know them collectively as the scalenes. Notice they are similar, but not identical in their locations. Let's take a look at it here first. What is the origin of the scalenes?
Transverse processes of C2 through C7.
Yeah, notice not the atlas up here, but C2, 3, 4, 5, 6, and 7 out here on the transverse processes of this. Notice this doesn't connect to the head, just to the neck region. And what is our insertion?
Anterior lateral surface of ribs 1 and 2.
There we go. So notice it is similar in its location, but not identical. And let's go ahead and go to our drawings and actually draw that. Let's see, where does my scalenes go? There it is on this one here. I need to move this over out of the way. This over here out of the way. Excellent. Again, some of these drawings don't do the best of showing us everything. We know it's C2, 3, 4, all the way through 7. So obviously I'm not seeing all 7 cervical vertebrae, but we know they're there, and that's a good reminder of what they are for our origin. And then our insertion, anterior lateral parts of rib 1 and rib 2. So those are our scalenes. All right, we've drawn the origins and insertions, but I actually like to look in at the picture of this muscle for the next part, because like I said, it is similar in its location to the sternocleidomastoid, but not identical. And so not surprisingly, we see similar actions, but not identical. Remind me again, how many actions does the sternocleidomastoid have?
3.
Great.
What were those 3 actions?
Flex head, elevate sternum. Rotate head.
Excellent. Flex head, elevate the sternum or the ribs. We could think of it that way. And what was the third one?
Rotate head.
Excellent. The scalenes also have 3 actions. What are the 3 actions of the scalenes?
Elevate ribs, flex head, and laterally flex head.
Excellent. Notice both flex the head, both elevate the ribs or the sternum. All right. So it also brings the head down or brings the ribs up for an inhalation. However, because the sternocleidomastoid attaches to the head, it can rotate the head. Whereas the scalene doesn't actually attach to the head, it attaches to the lateral aspect of the cervical vertebrae. So exactly, it laterally rotates the head. All right. God, I don't even remember the company now. I was trying to remember it. Back in ancient times, and by ancient times, I mean like the 90s, there were these commercials for, I think they were cassette tapes or a speaker system or something along those lines, where it had these little white dogs that would listen to their master speaking through the radio. Anybody remember the name of that company? Anybody have any idea what the heck I'm talking about? No, okay, fine, whatever. You're all young. I hate all of you. Anyway, I always think of this in terms of a dog. When you say his dog's name or he gets all excited, right? He kind of does that, gets all excited, brings his head up, tilts his head to the side to see what you're talking about, right? Hoping to go for a walk or something along those lines. So again, that's what we're talking about. Flex the head, elevate the ribs, and laterally flex. Oops, I rotates. Sorry, my bad. Laterally flex the head. Whereas the sternocleidomastoid rotates, this one laterally flexes. So you can see they're similar in their actions because they're similar in their locations. But their insertions and origins show you the difference of what it is that they do. All right. Questions on that? Here is a second muscle group on our head. Again, this muscle group is made up of 2 muscles, the zygomaticus major and the zygomaticus minor. But do we have to worry about distinguishing the individual muscles? No. We just need to know them collectively as the muscle group. That is the zygomaticus. And so when we look at our bones, what is the origin of the zygomaticus?
Zygomatic bone.
Zygomatic bone. That's convenient enough. Notice not the arch, the bone. We're talking about like the little apple of your cheek right there. That is the origin. What is the insertion?
Cannon muscles at corner of the mouth.
Yeah, remember, most skeletal muscles connect to bones, but most isn't all. In this case, it connects to the skin and muscles on the lateral aspect of the mouth. And of course, as we know, when a muscle contracts, it brings the insertion towards the origin. So what is the action of the zygomaticus?
Smiling.
Well, the activity that it would do would be smiling, but you get the right idea. It elevates the lateral aspects of your lips, what we commonly refer to as a smile. So yes, it allows us to smile, but the action is to elevate the lateral aspects of the mouth. Bring up our cheeks, bring up the corner of our mouth, which you're right, produces a smile. Questions on that? Notice, just that easily, we are now through all of the muscles that you are responsible for the origins and the insertions on associated with the head. However, there are still some muscles that, while we don't need to know their origins and insertions, we need to know their locations and we need to know their actions, what it is that they do. The first of this is our buccinator. The buccinator is a deep muscle that fleshes out most of the cheek. You don't have to know the origin and the insertion of it, but I will point out to you, it can be pretty easy to find on our models, on our charts, by some of the other muscles we're responsible for. Notice here is our zygomaticus coming up. And then coming down here is our masseter. And notice right here in the space between the two, this indentation between the zygomaticus and the masseter is where you find that deep buccinator muscle. So we see this really nicely here. And of course, well, let's bring it up. The other place you should, of course, be looking for all of these things are on the models. So if we go to Cosumnes River College's Muscle Anatomy, Motor Anatomy Lab, and will it be on the torso? There we go. That works. It's not the best view, but it'll work. This is not a best head model. It comes off of the torso, but you guys can see the head model here. Notice something like this could easily be on the exam. And notice, here is our zygomaticus going to the corner of our mouth. Here is our masseter going to the mandibular angle. And so let's change colors. This pinkish muscle that is deep right here, forming the cheek, that is our buccinator. So again, find that V of the zygomaticus and the masseter, and that will help you to find the buccinator. Now, as I mentioned, you don't need to know the origin and the insertion of it, but you do need to know its function. What does it do?
Press cheeks against teeth and lips.
All right, now what do you use that for?
When eating.
Yeah,
Pushing
exactly.
food.
Its job is going to be to squeeze the food, to present the food to your teeth, keep it there, stabilize in your teeth for mastication, right? When else might you use something like this? Any idea? Any of you play wind instruments?
Oh, I was going to say when you got a mouthful of water and you spit it at your younger sibling.
Absolutely, that's a good use for it. Absolutely, that is probably a good use for it as well. When you're playing a musical instrument, right? Unless you're Dizzy Gillespie, should you be puffing your cheeks out as much as possible as you're blowing air into it?
No.
No, you want to squeeze the cheeks. Now, some people often mention, for instance, like whistling. And whistling, true, you do have to squeeze the cheeks, but also when you whistle, you're puckering your lips. And remember, this doesn't pucker your lips. Another probably really good example of this is, of course, from the greatest movie ever made by mankind. What, of course, is the greatest movie ever made in the history of all of mankind? It is, of course, Zoolander. And what is Zoolander known for?
Deal.
His duck lips.
Yeah.
There you go. Blue Steel. There you go. Blue Steel, that squeezing of the buccinator, all right, for that beautiful look. There you go. Excellent. All right. We talked about circular muscles. Here we have one here, the orbicularis oris, helping to form the opening, protect the opening of the oral cavity. Right, so this is the one that puckers your lips to blow a kiss or to whistle or to, blow out your candles or whatever it is, something along those lines. Notice we also have a circular muscle that goes around the eye. Notice it's going around the eye. We're not talking about the elevators. I know that picture points it to the eyelids, but there are different muscles that move your eyelids up and down. This is really a circular muscle around the eye. Make a point of emphasizing that because, again, this is the one that doesn't allow you to just blink. We're not talking about just blinking. We're talking about closing your eyes, right? Like you're in the dust storm, right? You want to squint your eyes closed, or it's late at night and you don't want to wear your glasses. So you're squinching up your eyes to try to be able to change the curvature of your cornea so that you can read more clearly, right? That squeezing of the eye is what we're talking about with that orbicularis oculi. All right. Next, we have a two-bellied muscle. We see it on this picture, but let's also turn it this way as well, and we can see both bellies. This muscle is what is known as the occipital frontalis. It is also sometimes referred to as the epicranius. Epicranius is an appropriate name for this muscle, and I would totally be acceptable of that. I just don't like it because occipital frontalis, while there's more words in it, it tells you where both the bellies are. On the exam, I could have an arrow pointing right here, identify the muscle, be specific, and what's your answer to that going to be?
Frontal belly of the occipital frontalis.
Exactly. Or I can have an arrow pointing here on the exam, identify the muscle, be specific, and what would your answer to that be?
Occipital belly of the occipital frontalis.
There you go. Perfect. Excellent. And again, I'll have those magical words, be specific, to remind you that I'm talking about a specific identification. Notice we don't have to worry about the origins and the insertions for this, although it's pretty easy to tell. One sits on our forehead, frontal bone, one sits on the occipital bone on the back, and so that's kind of where they get their names. But I noticed one other thing about the handouts that I have. The handouts that I have at the bottom of the page, there's the muscles, the origins, the insertions, the actions, all of that. But at the bottom of the page, there are some bolded terms. And one of those bolded terms is the epicranial aponeurosis. Those bolded terms on the bottom of each page are not muscles, but they are structures you are going to be responsible for. And this indeed is one of those structures you'll be responsible. So on the exam, I could also have an arrow pointing here and say, identify this structure. And the structure is the epicranial aponeurosis. Remember, an aponeurosis is just a term for a broad, flat tendon. This broad, flat tendon happens to be on the top of your head, epicranial aponeurosis, right? And it connects these two bellies together. Because what is the action of the occipital frontalis?
Raise
There you
your
go.
eyebrows.
It raises your eyebrows, like in surprise. However, if you're like me and conveniently don't have a whole lot of hair on the top of your head, one of the things that you may notice when you elevate your eyebrows, your whole scalp moves. Right? If you put your head on your scalp and elevate your eyebrows, you'll feel it whole thing moves back and forth. And that is because these two bellies are connected by this epicranial aponeurosis.
So when you raise your eyebrows, you're triggering both bellies.
Yes. Yep. All righty. Last one, the platysma. The platysma is this big, huge, broad, flat muscle that sits over, and there are a tremendous number of muscles in the neck and in the jaw. And this kind of sits over the top of all of them. It's this big, huge drape, broad, flat muscle, superficial muscle over the top. And notice it actually comes up onto the mandible as well. Again, I make a point of emphasizing its location, not because you need to know its origin and insertion, you don't. But again, understanding the location of this can help you to understand its function. Obviously, being inferior to the mandible, not surprisingly, it depresses the mandible, brings it down. But notice, because it actually comes up onto the anterior surface of the mandible, when it contracts, what else can it do?
Out your lip.
Right, protrude your lower lip, giving you that pouty face. So we have this big, huge superficial muscle there that is the platysma. I think I have, perfect. Here is the chart in your textbook. And in that, you will see, I mean, not in the classroom, this big, broad, flat muscle that sits over the top. Notice they only put it on the one side because so that we can see all of the deeper muscles, including the sternocleidomastoid that are underneath it here. But here we get that great view of that superficial muscle. All right. And again, another way you can see this for yourself, it brings down the mandible. But if you clench your mandible with like the masseter and the temporalis, when you contract the platysma, right, instead of bringing the mandible down, you can put tension in it. Not so much that it brings your ribs or anything like that up, but you can get that kind of Hulk neck, that big, huge, right? You can feel that muscle in that angry neck that you get, that Hulk neck from that platysma. That is that big, broad, flat platysma putting that tension in it. All right. And there you go. Just that simply, we are through all the muscles that you are responsible for on your head. All right. And again, as we mentioned, the whole point of these are, of course, to woo women. If you think of the muscles we just talked about, right, you can use your orbicularis oris to, right, you see you're walking on a beach and there goes a Jonas brother. So you whistle at him, right, give him the old eyebrows, wink at him, smile, right, and when he keeps walking, then you're sad and you're angry. So there you go. All right. Questions on any of that? All righty. Excellent. That's how we're playing the game. Not tomorrow, but Thursday, we will continue where we've been on. We finished part 1 of the muscle contraction. We are going to do 2 and 3 together. Oh, not together, but both of them on Thursday. And then we are going to move down into the muscles that control our trunk. So that's the game plan. The same thing we did today is what we're going to be doing for the rest of this section. Muscle physiology and lecture, and then origins, insertions, and actions in lab. All right, questions on that? All right, it's a little bit early, and even though we have a lot to cover, we did a lot today, so I think this is a good stopping point. We'll go ahead and take a break now. I will remind you, you have 4 days off before the next exam. Tomorrow, Saturday and Sunday, and next Wednesday. That's it. Between now and the next exam, there's only 4 days you're not being presented with 4.5 hours of new information. Take advantage of this time. This is one of your big study days coming up tomorrow. So again, look at the stuff we've covered and start getting ahead of this material so that you are prepared. Now you've seen how we're going to do it. Now you see what's expected of you. Now you've seen the resources that you should be doing to take advantage of. So make sure you are using, heck, the rest of today, but also tomorrow to the best of your advantage. It is really, really important that you guys start bringing up those exam scores. All right. Like I said, 3 things that need to be fixed. Read the questions carefully so that you answer the question that's there. Answer all parts of a question. But as we saw today, I'm looking for detail. Don't just tell me that calcium enters the synaptic end bulb. Tell me that the neural action potential depolarizes it to threshold, which opens voltage-gated calcium channels, which allows calcium to enter the cell. I want precision. I want details. I want specifics. Which reminds me, I also will release your exams now as well. So I will go release your exams so that you have those to view so that you can prepare from there. All right? Any questions on any of that? All right. Use your day off well, and I will see you guys on Thursday.